All right, so let's get started. It seems like people are starting to slow down in terms of coming in. So hello, welcome to our winter 2024 CCHAB meeting. Thanks so much for coming. So we're going to start with some quick introductions of our current CCHAB co-chairs. And I'll start. I'm Karen Odkins. I'm with the Freshwater and Estuarine Harmful Algae Bloom Program. And I'm the lead for CDFW, the California Department of Fish and Wildlife. And Sarah, do you want to go next?
Hi, everyone. I'm Sarah Ryan, the Environmental Director and Emergency Management Director for the Big Valley Band of Pomo Indians.
And Jamie?
I am Jamie Smith. I'm a research scientist at the Southern California Coastal Water Research Project on harmful algal blooms.
Thanks. All right. So we have a super exciting agenda for you all today. It's our annual kind of extended meeting. We're going to go until 2 p.m. today. So we're going to get lots of status updates today as the kind of first meeting of the year from the statewide FHAB program, from the water board's regional coordinators, and from our CCHAB co-chairs and other entities that wanted to give an update. We also have some really awesome science talks that I'm excited to share with you and programmatic updates. So thank you all so much for attending. Some quick announcements before we really jump in. So the CCHAB co-chairs, we're trying to think about the best way to do this meeting in the future. And so we're going to be sending out a survey in the coming weeks that will kind of ask you for your vision for the future of CCHAB. So keep an eye out. We'd love your opinions. Does anyone else have any quick announcements at the top of the meeting today? Anything you want to share? Okay, great. Well, we can also, if we have extra time at the end, if people have things they've thought of, we can do it then as well. Some quick housekeeping. Please remember to keep your speakers muted if you're not speaking, and hold questions until the end of the presentation, and then raise your hand and we'll do questions there. So without further ado, let's jump into our first item. I don't know if Carly or Marissa, who's presenting for the FHAB program update, but if you want to share your screen, I'll stop.
Yes, hold on.
Okay.
I gotcha.
Thank you.
Okay. Hi everyone. Thanks for having us today. My name is Carly Nilsson. I am the co-lead for the State Water Board Freshwater and Estuarine Harmful Algal Bloom Program housed out of the State Water Board. And we're just going to give you a quick update as to what's happened this year. Just to orient folks who may not be as familiar with our program, we started in 2021 based on an Assembly Bill 834 that passed, and it allocated us 5 staff and some contract funding for lab work and things of that nature. So as you can see, our 5 dedicated staff are here in green, but there are a lot of names here that aren't in green and black, in fact, and those are our FHAB coordinators throughout the state. They're often surface water ambient monitoring program managers, but they're borrowed and volunteered to help us with a lot of our HAB event response monitoring and communication with the counties for posting. So we are eternally grateful for the borrowed help we get throughout the field season, because as you can see, 5 staff isn't that much for an entire program. And below here is a link to see all of those staff and their contact information, and we keep that pretty up to date. Some other resources that we house on our portal. So we actually have a map. Most of you have probably seen it multiple times, hopefully. And on that map, we put on there any HAB events that we receive. So anything that we hear from the public, agencies and that nature, we post in near real time, usually within 2 business days, and any corresponding advisory information. We also have information on there for the public and different counties to borrow as to how to stay safe, including FAQs and different things, especially for pet owners and things of that nature. We also have information as to interpreting our different advisories, because they are based on trigger levels. So folks know what to be aware of with these different colored signs and what they should be thinking about. And then as well as some information, so we have some consistent and reliable information coming to us. So we have standard operating procedures for collection and reconnaissance and things of that nature to make sure that we're having consistent data collection throughout the state and folks know what they should be doing. Okay, so now I'm going to talk about our holiday assessments. They're kind of one of our big bread and butter monitoring programs throughout the state. In terms of they've been most consistent. They started in I think 2017, so about 7 years now that we've been having them. And it's a great opportunity because it kind of gives us a twofer. So we get monitoring data throughout the entire HAB season on a water body, as well as information to help inform the public as to any recreational safety concerns. So just to give you a little bit here as to some of our stats, we conduct these assessments generally pre-Memorial Day, pre-4th of July, and pre-Labor Day. We've sort of shifted a little bit. So for those water bodies where we're not really seeing HABs in the late May season, some folks have opted to then sample actually post-Labor Day, since sometimes our summer season, HAB season, can last quite a bit longer than the Labor Day assessment. So the big aim is just to make sure that we have 3 consistent sampling events throughout the HAB season. Here we have marked, I just kind of cataloged our main ones. So we had, as you can see, like more sites evaluated with each holiday assessment. And I'll go into more detail in the next slides. The one thing I do want to highlight is that, oops, I'm going backwards, or no, I'm going forwards. Okay, the one thing I do want to highlight with this is that with all of these numbers that we get, often these samples are collected by those FHAB coordinators with that map I showed you earlier. So we are very reliant on them for the boots on the ground and helping us get this like pretty robust data set throughout the field season. This slide is just to reorient everyone as to our advisory schematic. This is focused on our planktonic, but we did collect samples for benthic as well for our holiday assessments. And this is just to highlight that we still follow our regular response protocol with all of these samples. So any samples that are collected, especially if folks have visual observations on the ground, then they update the map immediately, showing that there was a visual observation. And then when we get that data back from the laboratory, because those samples are all sent to the lab for toxin analysis, then we update the advisory based on our different trigger levels, and then do that communication to the counties for posting and make sure that that loop is completely closed in that regards, to make sure that we're getting the information out to the public as quickly as possible. So I just wanted to share some numbers as to the advisories we had with each one of these holiday assessments. As folks know, 2023 was a fairly wet year, so it was pretty quiet earlier in the season. We had 69 sites sampled. And as you can see, we had not too many advisories that we had to put out. More samples were collected for pre-Independence Day, and then we had started to see, we had 1 danger advisory, an uptick a little bit in the caution advisory. And then if you look at the pre-Labor Day, you can start to see that things started to start blooming a little bit more. So a few more cautions, some warnings, and quite a few more dangers, and then as well as a pretty big uptick in our toxic algae alert signage. And I'm not going to go into too much detail because we're going to have some updates from the FHAB coordinators after me. So they're going to give a pretty good, robust update for each one of the holiday assessments for their different region. And with that, I'm going to hand it over to Marissa Van Dyke to go ahead and discuss a little bit more about our partner monitoring program.
Hey folks, good to see everyone here at CCHAB today. So yeah, I'm just going to continue the update going into partner monitoring, and just again an announcement that we are soliciting for projects for partner monitoring with our FAB coordinators. So if you represent a waterway, a recreational area that you're of interest of having monitoring done, you can and you're interested in collaborating, perhaps putting the boots on the ground, that kind of thing with us, please reach out. And you do next slide, please. Here's a big old QR code. We're trying to integrate those in a little bit more so we can quickly share our information with everyone. So if you're interested in partner monitoring with that FAB program, you can reach out to our general inbox or contact your regional board coordinator. as we're getting these projects up and running for the 2024 year. And also with these partner monitoring projects, oftentimes sampling supplies are available. You know, it's case-by-case basis, that kind of thing, but it is an element of support as well. And some of those partner monitoring projects have included, you know, doing routine, kind of weird prompt here from Zoom. Sorry, it includes routine monitoring throughout the recreational season at your sites. It can include planktonic monitoring, you know, depending on, you know, what kind of blooms you may be encountering in lakes or in streams. Also, we cover projects with benthic monitoring as well. Let us know if you're interested. Next slide. All right, some other updates. So for each year, we've been always tallying the number of reports that are submitted to our reporting system. Again, this is an online website with a report form where we can capture photos, that kind of thing. And these are what are voluntarily submitted because, again, there are no regulatory requirements. So we've got our numbers for this past year. And we did see less reports coming in. Particularly, we saw, you know, a later season for the blooms, particularly in lakes and streams, likely because of the wetter year. And also, we had a little less funding for our partner monitoring, so we saw a little less routine monitoring at some of our hotspots in previous years as well. So we really encourage, if you have the ability to do some component of the monitoring, you know, boots on the ground, lab analysis, supplies, that kind of thing, please reach out and we can see where we can fill in those gaps. Next slide. Also, this past summer, and you know, it's always hectic in the summer, so we wanted to bring this up again, that our satellite imagery tool that's hosted just for California, and it presents the pre-processed satellite imagery from NOAA and the federal agencies for California and the resolvable lakes and wide channels. So we've got the cyanobacteria index up and running, you know, for a number of years. I want to say 6+. And over the summer, we were able to add some additional features, including presenting chlorophyll A for the resolvable lakes and channels. So you can toggle between seeing chlorophyll A remotely sensed and the cyanobacteria index, either separately, or you can also graph those metrics together for in an individual waterway. So that's been really handy seeing the production there. And an additional component that we added that was normally on a request basis was also displaying one-day data sets. So instead of the composite data, which is a 10-day composite normally, you can toggle between the 10-day composite for both metrics or 1 day. So the 1 day is helpful for doing field assessments. So you can see like what side of the water body has, you know, higher density, that kind of thing. So you can see just the prior day of the satellite imagery, which is helpful. All right, next slide. And also we, in the last couple of weeks, we were able to publish our annual report for 2023. This is for our entire program, and it's part of our legislative reporting framework as part of the Assembly bill that established our formal program in 2019. There we provide an overview of the different, how we work towards meeting the different objectives of the Assembly bill, ranging from incident response, monitoring, research and development, and education, outreach, and data management. Please take a look. And that also we highlight gaps in the program since it was not a fully funded program at the onset. So we are still seeking additional resources to fully implement that Assembly bill. And you can find this report here on the QR code, and also previous reports as well. All right, and that's the end of our update. Thank you, folks. And if you have any questions for the statewide FHAB program, please let us know. Otherwise, we'll hand it off to each of the regions. Yes, and thank you. Randy Turner from SFEI. He's the primary lead scientist on the satellite imagery tool that we just spoke to. And we're always seeking feedback from the users, the user group. So please reach out to him, or again, CCHAB reports for any inquiries, requests for additional features. Also, if you want to connect to our API. Thank you. I'll hand it back to Karen.
Thanks, Marissa. All right, well, we'll get started with our regional updates now, starting with, sorry, Mike, region 1. Mike, are you here? Okay, cool.
Yep,
Thanks.
I'm here.
You can go ahead and
are you gonna share my?
Do you have yours? I figure people might have done last minute
Yep,
updates.
I do.
But, okay.
Okay, can you see that okay?
Sure can. Looks good.
Okay, great. Hey, good morning, everybody. My name is Mike Thomas. I am the FHAB coordinator in the North Coast region. I work closely with Rich Fadness, who is our swamp coordinator. So just giving you a 2023 season recap. Oops. Routine monitoring that occurred in the region. Klamath Basin, we had the Hoopa, Karuk, and Yurok tribes, as well as Pacificor and US EPA conducting biweekly monitoring. In that graphic to the right there, you can see a map of the basin, sampling stations. We also had routine monitoring in Big Lagoon, conducted by Big Lagoon Rancheria and US EPA, again doing biweekly microsystems monitoring. Also in the Russian River, we had Sonoma Water and Sonoma County conducting some visual assessments in cyanobacteria ID. So jumping into the routine monitoring in the Klamath Basin a little bit, these two graphs here show Copco and Iron Gate reservoirs. On your vertical axis, you have microsystems, and on the horizontal, your date. Sampling occurred at 2 public health stations in each reservoir, and you can see around that mid to late August timeframe, microcystin concentrations increased and led to some danger postings. Moving downriver of the reservoirs, we have various Klamath River stations. Again, this is just showing towards the September timeframe in the season, later in the season, there were some caution postings. And also want to note that the Yurok tribe issued some level 1 and 2 advisories in the lower river. Big Lagoon partner monitoring. So as Marissa was describing, this is part of their partner monitoring program. In addition to their routine monitoring that they were doing, we also collected a sample for cyanotoxin sweep analysis and cyanobacteria identification. As you can see in the graph here, we largely saw microcystins, which were driving the caution and warning postings this season. And interestingly, we did document 6 different cyanobacteria genera in the lagoon throughout the season. Pre-holiday assessments, we did opt to do most of the later season sampling by including that post-holiday sampling event. Here are the water bodies that we monitored and the entities that did the monitoring. You could see we had a caution in Lake Pillsbury towards the later part of the season, and toxic algae alerts in Lewiston Reservoir and Salmon Creek. So special studies, we continued our benthic monitoring in rivers. This is implementing that tiered approach that we keep recommending. This is deploying SPATs as sentinel samplers. When we start seeing increasing concentration in those SPATs, we'll then conduct visual assessments to determine percent cover and then collect benthic mats to confirm bloom toxicity. So we did this weekly in the Eel, South Fork Eel, Russian, and Navarro rivers from mid-May until mid-October. And results from these studies are being reviewed by the benthic subcommittee of CCHAB when revisiting their current guidelines. So quickly, some results from that. In the South Fork Eel River, In this graph, we have cyanotoxin concentrations in spats and mats on the left vertical axis and cyanobacteria percent cover on the right vertical axis. And this is all over time. Spat concentrations are in the solid lines, mat concentrations are in the dashed and open circles, and percent cover cyanobacteria is in that dotted line. Basically, what we're seeing is that we're seeing those increases in spats, which correlate closely to increases in benthic mats, and those increases are occurring prior to major increases in cyanobacteria percent cover. So we did issue that toxic algae alert in that mid-August timeframe. But kind of to the contrary, in the Russian River, you can see here that spat concentrations remained kind of low throughout the season. And when benthic mats did appear, they were very low in concentration. So no advisory was recommended at this location. Some studies we did with US EPA, we did participate in that ROAR project in developing standardized benthic sampling protocols. This basically involved a lot of transect and disturbance sampling in the South Fork Eel River. We also assisted with some RNA and DNA research, looking at upregulation at different growth stages using paraphytometers, which is that equipment you see in the bottom right here, and doing some sequencing of ANA-F genes. Some other research, we were collaborating with the University of Nevada at Reno and CSU San Marcos, looking at spatial heterogeneity of benthic cyanobacteria and cyanotoxins. And this was in the Salmon and South Fork Eel Rivers. We were also able to participate in our helicopter surveillance program at the North Coast region and got up in the air and were able to identify easily benthic cyanobacteria mats, specifically microcoleus in the riffle habitat. Trainings, we hosted the US EPA ROAR training here in Santa Rosa on the Russian River. We also participated in the Blue Lake Rancheria and Wiat tribe trainings in early August, then hosted a series of virtual and field trainings in late August for our North Coast partners. And posting and illnesses, I'm not going to go into a lot of detail of what these tables are saying, but benthic and planktonic postings. Interestingly, we did have a marine bloom this year in Humboldt Bay, which is that orange bloom you see there to the right. There was an illness associated with this, although the bloom was not toxic, and that's likely due to bloom byproducts such as ammonia exposure. And then we did have one human illness in the Russian River. And all that work could not have been done without the help from our partners, so big shout out to them. And I think that is all I have.
Thanks, Mike. Any quick questions for Mike? All right. Region 2. I think Christina.
Hi. Yeah, let me
Great.
share my screen. Let's see, where is it? Can you see my screen?
Yep.
Okay. Can you see my notes, or is it just the presentation?
I see the PowerPoint window. It doesn't look like it's in full screen mode yet.
I don't know how to do this. Let's see here.
I think there should be a little presentation.
Yeah, I pressed the little button.
Oh, you did?
It's
Okay.
not working. Yeah, it's showing up on my double screen, but I guess you're not going to get to see that.
Yeah, maybe
Okay,
it's the other window.
I don't know. Nope. Well, can I just go from here? Is it
That's fine.
big
Yeah.
enough? Okay.
Go for it.
I'll just start over again. Okay. Hi, Christina Yoshida from Region 2. It's always so hard to go after Region 1. we aren't as productive as they are, but thanks for having us. So
Christina, sorry to interrupt you.
Yeah.
It's a spreadsheet now. You
Oh.
had the PowerPoint. I think you were right the first time. I could see your PowerPoint slide.
Oh,
How about now?
No, it's black.
No? How about now?
Okay, now I see a PowerPoint. Just do
okay.
slideshow.
Oh, mine must be
Yeah,
delayed.
there you
I'm
go.
seeing something else.
Is that better?
Yep, we're there.
All right. Okay,
Awesome.
cool.
Thanks,
Oh my
Emily.
God, I
I'm
hope you didn't see
I'm
anything
delayed
weird.
here.
Okay, so where was I? All right, so incident responses. We had 16 incident responses throughout our region. 8 of them were confirmed HABs. It was spread across 5 counties. It's kind of exciting. We got to interact with 6 new agencies. So trying to develop those relationships so people will reach out to us when they see something weird. There were 3 sicknesses reported, but only 1 was actually confirmed related to a HABs. There were 2 dogs and 5 wildlife, although wildlife was more due to the red tide event. But yeah, so we had 6 red tide reports and responses that we had in May and then in July and August. And of these incident responses, we did site visits 6 times. I won't go through this, but this is our table of where we were in case you care about the different water bodies and what happened there. We took part in the holiday assessment at 1, 2, 3, 4, 5 sites. These are the swimming areas that are not included in some of our bigger parks that are managed by like East Bay Regional Parks. Only 2 of them resulted in caution advisories, which is great. This year we're going to switch it out and do some different sites kind of based on these not having issues at a lot of these, but also developing some new relationships with partner groups. Speaking of partner monitoring, this year we worked with the Rotary Nature Center at Lake Merritt. So if you recall, a couple of years ago, we had the red tide event and the huge massive fish kill in Lake Merritt, which is in downtown Oakland. It's a very urban lake, very visible, a lot of people interacting, well, not really in the water, but around the water. So we worked with the community group and we gave them funding to do monthly sampling from May to October at 2 locations. We also loaned them one of our sondes to do weekly DO readings with community members. We also did 2 training events with a group in Napa and Sonoma and gave them equipment so that they can help respond to incidents. There's only 2 of us doing HAB work, less than 5% of our time. So we really need as much help as we can from these groups. And also, I just wanted to give a big shout out for East Bay Regional Parks. I know there's a few of them here today. They really collect a lot of data. So every week they're at their major lakes and they share their data very timely, and we're able to post that to the HABs reporting map. And then lastly, again, we had another red tide event. It was much more subdued than 2 years ago when we had a big fish kill off, but there still were some deaths. And in response, because it's just a lot more complicated than responding to streams and lakes and whatnot, our group got together with our nutrient management team through SFEI, the San Francisco Estuary Institute, to develop a communication and incident response strategy. They're in line with our state procedures, but it just kind of adds more to it since it's so complex and expensive to go and take samples out in the bay. So we are working on finishing that up for the 24 bloom season. All right, that's all I have.
Thanks so much. Yeah, and East Bay will be giving their own update at the end as well.
Great.
Yeah, thanks so much. So Region 3, I think we got Melissa. Is Region 3 on here? All right, maybe not. We can come back to them at the end. I'm not sure. Marissa, had you heard anything from them?
No, I haven't. Doesn't look like Melissa Daughtry's online. We can check with her.
But we can
quite
give
a bit of monitoring though. Oh yeah, you want to fill in?
Yeah, we can do a
Okay.
quick update. So you might have
Thanks.
noticed from the slides that I was showing earlier that Region 3 did have a few dangers, San Antonio, and I think Oso Flaco for the lake and the creek. So they're once again a region that doesn't have a dedicated FHAB coordinator. So they borrow from their swamp program to the best of their ability, but they have a very robust swamp monitoring program. So they try to just leverage when they're already going out for that program. So they do participate in the holiday assessments, and they sample 10 sites for those 3 assessments, and they contribute half of the funding so that they can make sure they hit those 10 sites regularly every single year. So that is their consistency, is at least providing that long-term data set for those 10 sites.
Thanks, Carly. That's awesome. Great. Region 4. Do we have Emily?
Hi, yeah, this is Emily
Thanks.
Duncan with the LA Water Board. We also don't have a dedicated FHAB person, so I cover that in addition to some other programs like Swamp. But just to give a brief overview, I had about 8 incidents reported through the portal or through the system in 2023. Besides Pyramid, Lake, and Castaic, which are monitored by DWR. So kind of 2 of the larger lakes in our region get regular testing and monitoring, and then caution warnings as required. And usually those lakes do have some level of caution every summer, especially Pyramid Lake. A couple of them, I did one site visit for Arroyo Seco, which is a river stream in our area, just did a visual inspection. And a lot of the issues that I have with the public reporting is that folks don't provide a way to contact them, and it's anonymous. And so there's not a lot of information. And then when I reach out to park managers, be it through the city of LA or the county, typically there's not a lot of response or information. So it's a little bit tricky developing those relationships. And then we did have a red tide event that was reported at Manhattan Beach Pier. I believe it was red tide. And a few, I think 2 or 3 of the notices I got were for like stormwater retention basin or locations that didn't have public access. So there was one private lake, and that happens a lot in our area as well. So there's no public access, there's a fence around it, and the person reporting doesn't have access to the water body. And they were complaining about smell, and they were complaining about potential HAB, but again, unless somebody that lives in that community is going to report it and provide us access to like do a visual assessment and take a sample, then we don't really have a way to check. And so those are a couple things that tend to happen. But yeah, I was going to start emailing folks, water body managers like Lake Piru. Jamie Smith at SCORP did a large survey of our freshwater bodies. And some folks like at Lake Piru kind of do their own monitoring, which is great for them. And then based on her work, we also put together a pretty decent list of contacts. So every year I'll send an email out, but the interest in holiday assessments has been kind of low. I think folks might be more interested if they could do some sort of in situ quick test. I don't know, that's something that maybe would garner more interest in our region. They could do like a dipstick type of situation and then decide if they need to send a sample off for analysis. But yeah, those are just some observations that I have. And what's cool and upcoming is that we're working with Jamie on a coastal study. It's being leveraged with the BITE 2023 sampling in Southern California. And so hopefully at a future CCHAB meeting, we can give an update. JB and I can talk about our coastal hubs project. Yeah, and that's it. Thanks.
Thanks, Emily. That'd be great to hear for updates as they come. Region 5, Dana.
Hi, I do have some slides to share.
Thanks.
Okay, are you seeing Are you seeing the slides?
Sure am.
Okay. Hi, I'm Dana Schultz with a freshwater hab coordinator with the Central Valley Regional Water Board, Region 5. Let's see if I can get this menu out of here. Okay, so for our year in review, I have updates on our event response efforts, some contributions to the HAB reports map, updates on holiday assessments, and 2 partner monitoring projects we have going on in the region. So for event response this year, we received 36 bloom reports across 17 counties in Region 5. 64% of those reports came in from the public. The majority of those were through the Water Board's report form, and a couple were through the poison control hotline. And then 36% of the reports that came in were reports from other agencies that might submit a report when they're out sampling and see a suspected HAB. And of those reports, the HAB illness work group took on 15 cases from our region this year. And they determined that 6 of those cases were HAB related. So in our region this year, we had 4 human illnesses, 1 dog death, and 1 fish death event. And then when we do the event response, we figure out the appropriate advisory and post that to the Water Board's HAB reports map. But I also wanted to highlight that there are multiple monitoring programs in Region 5 that voluntarily share their data with the FHAB program so that we're able to post the appropriate advisories for that data on this resource for the public as well. So I just wanted to shout them out and say thank you for sharing the data. And then for holiday assessments in our region, we were able to sample and worked with people to sample 5 water bodies. So Hensley Lake, Eastman Lake, Clear Lake, Kauai Lake, and Kauai River. And the sampling agency is listed in that table. So at Hensley Lake, we did detect toxin and issued lake-wide caution advisory. At Clear Lake, toxins were detected and advisories were issued, and that data, you can see it with Big Valley Band of Pomo Indians with their data set. And then luckily this year at Eastman Lake, Kauai Lake, and Kauai River, we did not detect toxin during the holiday assessment sampling, so no advisories were issued. And I'll note that at Kauai Lake and Kauai River, flows were so high this year, the sites weren't accessible or safe to sample until post-Labor Day. So we only had 1 event with them. And then moving into partner monitoring, for a couple of years now, Region 5 and State FHAB has worked in partnership with Restore the Delta, based near Stockton. And at right, you can see Restore the Delta staff and some of their interns out sampling the waterways near Stockton. So this year in 2023, Restore the Delta, Spencer Fern and his group of interns sampled 7 sites in the Delta. They sampled between June and September. We sent in samples monthly for microcystin analysis with ELISA, and they also conducted the rapid strip tests weekly as a screening tool. And the results from this year, so there was not a lot of microcystin detected, so strip tests all came back as non-detects. And in the ELISA samples that were sent off monthly, there was only 1 sample from 1 site at 1 date early June that had microcystin detection, and the levels were below what would warrant a caution advisory. And we're hoping to continue this partnership, and Restore the Delta is planning to continue sampling in the coming summer. Another partner monitoring program happening in R5 is with state parks. And this year, this partnership was led by State FHAB. So in partnership with State Parks Goldfields District, their staff sampled 5 sites along the North Fork American River. They were able to go out 4 times for sampling between July and October. They would collect water grabs every time and algal mat samples if they observed the mats. And so in those 4 times they went out, anatoxin A was detected at least 1 site and 1 sample during each visit. The concentrations of anatoxin ranged from 0.16 to 0.62 micrograms per liter. And then I also just wanted to highlight some other work being led by other folks at R5 related to HABs. So we have a number of projects going on in the Delta. Later in this meeting, you'll hear a talk that our 5 folks helped out with about microcystin concentrations in clams and crayfish. There's a project looking at overwintering cells. And then there's also work focusing on the Stockton waterfront in 2022. And I'll shout out the little clip of the paper there to the right. So the lead author was Ellen Priest, and you'll see Janice Cook from Region 5 is on the author list as well. So definitely check out that paper if you haven't already. Another project at R5 is a study of Hensley and Eastman Lakes comparing HABs and nutrients. So that had folks from the Rancho Cordova office and the Fresno office contributing to that study. And recently, Stephen Huey gave a talk. The title's in the picture at the bottom right at the, I believe it was November CABW meeting. So I think there's a recording of that if you want to check that out. And then we also have a couple ongoing Clear Lake projects looking at a continuation of a driver study and ongoing work of the nutrient TMDL. And now we're working on some mostly data management tasks and planning for 2024. So I'll wrap up with that.
Thanks, Dana. So much going on. Now on to Sarah Ryan with the Big Valley Band of Pomo Indians for a Clear Lake update.
Hi, folks. Let me go ahead and share my screen. Basically wanted to show you our tracker for our sampling events. And I'm just showing you basically our summer into the fall. We actually had the highest toxin level for microcystin happened in November on the lake. So I'm showing you basically June through November, our sampling. And of course, this is the caution warning danger is based on the numbers, the criteria that we're all aware of, that danger is anything above 20 micrograms per liter. We did not have the anatoxin detects like we had in previous years, but we did have a higher toxin level. It was almost 17,000, I believe. Let me just pull that up. Yeah, almost 17,000. And that was higher than the last, certainly in 2022. But in 2021, we had 160,000 micrograms per liter of microcystin in the same area of the lower arm. So, so this year was a little bit higher compared to the previous year. Another thing that we do in our monitoring, so we're out there every 2 weeks. These are all shoreline sites, except for we've got some partner groups in the area who are going to take samples from the interior of the lake and bring them back to us. So we're out there. We do have some samples from Blue Lakes from that Mike spoke about, and then also some UC Davis sites. But we do look at we are tracking things like the number of the times that each of the sites had caution warning or danger, so that we can, basically show, is it all of the time that these sites are at caution warning and danger, or half the time? And quite a number of the sites that we have that are used for recreation, tribal uses, cultural uses, fishing, things like that. There's quite a number of sites that, more than 50% of the time in the summer, or basically this window of June through November, is above or is at caution warning or danger levels. As I mentioned, in terms of toxins, the highest that we saw was almost 17,000 for microcystin. I don't believe we got any detects of anatoxin throughout the whole year. Our highest site was at Jago Bay, which is in the lower arm. And so the toxin levels we are seeing actually sort of a mix of both the oaks and lower arms of the lake having approximately the same number of caution warning or danger. In terms of microscopy, we're also doing that and starting to look at nitrogen fixers, like when those are coming in. In this spreadsheet, the nitrogen fixing cyanobacteria are dominant in the, so we're looking at dominant and secondary cyanos, just to see, what kind of trends are there, and to understand the drivers a little bit better. So we are seeing nitrogen fixing cyanobacteria dominant in the earlier part of the summer, but as the summer progresses, I have a graph right here. As the summer progresses and the nitrogen fixers drop, we're seeing an increase of the microcystis, and then as that drops, a rise of cyanobacteria that's not microcystis or nitrogen fixers. So just something that we're tracking. And I've not done this for the other years, but we are in our 10th year of doing this monitoring on Clear Lake of basically the shoreline. And we are also collecting water chemistry and you know, basically habitat, or not habitat, but just observations of the bloom size. The other thing that we're doing on Clear Lake in the last couple of years, or in the watershed, we've got doing spat bags, or basically benthic sampling and observations, and that's through another program, but because we're looking at the impacts of cyanobacteria and cyanotoxins for drinking water systems, private drinking water systems. So we have been installing spat bags to basically collect the dissolved cyanotoxins that are floating by in creeks during the early season when the creeks are running. So that's usually in the spring. We had 8 creeks that we sampled last year, 2023. We were getting anatoxin and saxitoxin detects in the spat bags. We also did confirmatory grab samples at the same time, which included parts of the benthic mat that were present, and we were not, you know, able to capture that we were saying higher levels of cyanotoxins in the spat bags versus the grab samples. So we're continuing to, we'll be continuing that program in the creeks of Lake County. We did participate. There's an ongoing messaging meeting that the county runs, which is basically talking through the signage issues and communication to the public, as well as, you know, there were some human illnesses and a dog death on the confirmed on the lake this year. So we just are, all the partners are staying in touch regarding those issues. Also, finally, we do, Big Valley has the data and a map on our website that we need to update since the season is over, as well as a Facebook page called Clear Lake Water Quality, where we put pictures of the microscopy and just do a lot of outreach to the public to educate them about the issues and also the importance of nutrient management, because that goes hand in hand. And I think that's it for our part of Clear Lake. Thank you.
Thanks, Sarah. Quick question in the chat for you, Shannon had. So could you reiterate what the 2022 and 23 microcystin highs were? Oh,
Yes, so in 2023, it was 16,821 micrograms per liter for microcystin. And then let me look at 2022. Sorry, it's just taking me a second. 2021, I know it was quite high. So 2022, it was 790 micrograms per liter, and that was at Sulphur Bank Mercury Mine. So that was in the Oaks.
Okay.
Oaks Arm. Yeah.
Thanks.
Thanks, Shannon, for the question.
All right. On to Region 6, Sabrina.
Hi, can you hear me?
Yep.
Okay, all right. I always struggle changing from Teams to Zoom, so.
I know, we all do.
Okay, I'll share my screen. Are you seeing my PowerPoint?
Yep, looks good.
Perfect. Okay, I'm Sabrina Rice. I'm the harmful algal bloom coordinator in Region 6. So we had this many advisories this season. So this is just accounts for like the highest level any water body reached. And so we had 3 dangers and 11 cautions, 0 warning. Here's a map of just kind of where
they were. So our region is everything kind of east of the Sierras. I'm not sure if you can see my cursor, but Lake Tahoe here is kind of in the middle. And this is just the top part of the region, so you could see the words a little better. I know it's a little blurry, so I apologize. But the danger was at Indian Creek Reservoir here. And then here's the southern portion of our region. Crowley Lake was another danger, and then Silverwood Lake was the last danger. And Silverwood Lake is monitored by DWR routinely, and they share their data with us. Here are just some photos. Sorry, is this in the way? I always think it's just fun to see the different visuals that we're seeing out there. Indian Creek Reservoir did reach the highest level of toxins we saw in our region. I think it was around 400 micrograms per liter of microcystin. And typically we do see more anatoxin in our region, but this year it was almost all microcystin in all of the blooms we saw across the region. And yeah, I think these are just kind of cool to see. Here is a table. I just thought it was cool to compare the different kind of how much we have sampled since 2018 and the different advisory levels we saw, as well as the number of illnesses reported and confirmed to be HAB related. So, you know, we have kind of gone up and down on how many actual physical sample bottles we've collected. Last year we had a little more funding and just kind of like some interesting things going on in the Tahoe Keys. So we definitely sampled a lot more last year than we did this year. And, you know, the water level was much higher this year. So it is kind of interesting seeing, or sorry, the water level rose this year. We had a lot more precipitation. So it's interesting seeing that we had like a decrease in the number of caution advisories and danger advisories. We did have more, we have never really had fish kills reported in our region, and we had 4 reported this year. None of them were confirmed to be HAB related, but it was just kind of an interesting change in the season. We had 1 dog death confirmed to be HAB related, and that was in Lake Tahoe at Regan Beach, and then 1 human illness, and that was at Crowley Lake. For the type of monitoring we conducted, you know, we assess new and recurring blooms. I did not get a number on how many blooms we were reported this year. Everybody else seemed to do that. And then incident response as well. Special studies, we just continued the Red Lake monitoring this year. We previously had a few other special studies, but we decided not to do those this season. Just due to some changes in the monitoring and some other just projects going on in those areas. We did start a new partner monitoring program with Inyo County Environmental Health, and they were monitoring Diaz Lake, Mill Pond, Pleasant Valley Reservoir, and Owens River, just where Pleasant Valley Reservoir exits. And then we had the pre-holiday assessments, and we definitely focused most of our efforts on pre-holiday assessments. And here's kind of a table just of how many water bodies were sampled for each. We noticed in the last year that there were much higher hits during a post-holiday assessment. So we did kind of alter this year again to do most of our monitoring instead of during Memorial Day. We did it for the post-holiday assessment. And you can see here the different advisories that occurred due to those assessments. So we are seeing most of our higher numbers in Labor Day and post-holiday. And then all of our partners, we have definitely a very long region, so it takes a lot of time to drive throughout the whole thing. So we rely very heavily on our partners. I cannot monitor all those water bodies during the pre-holiday assessments without all of you, so thank you very much. And we had two kind of new ones this year, which was Spring Valley Lake Association and Green Valley Lake Recreation. We had previously worked with Green Valley Lake Recreation, but they didn't help for like 2 years, and then they came back again this year. So that was great to have them back. And Spring Valley Lake also does some monitoring on their own. So it's cool to partner with them and just kind of combine our efforts. And next steps, we're again always looking for new partners to expand our pre-holiday assessment so we can keep that data set growing. And we're in the process of, figuring out what type of special studies or monitoring projects we will be doing this summer. So more to come in the future. And that's it. Any questions?
Thanks, Sabrina. Not seeing any in the chat right now. So let's move on. Region 7. I think I didn't see Jeff on here.
Yeah, hey Karen, this is Marissa.
Awesome.
Carly and I can provide an update for Jeff, and this is the Region 7 high desert area. So Jeff Gerasti, he's the swamp coordinator and also fills in as he has time for HAB program coordination. Again, he's not funded, though he was able to sample during the holiday assessments for 3 points in time throughout the year at the Salton Sea. So Salton Sea spans Riverside County and Imperial County. And he monitored 2 of the recreational sites there, Bombay Beach and a state recreation area that resulted in caution advisory for those sites. And also he was able to sample at Weist Lake, and that's in Imperial County, and that resulted in a caution advisory as well. I believe they also had a fish kill at that site. We're again working with the county on potentially getting a monitoring program set up. And another effort in his region has been a coordination with the US EPA Office of Research and Development. So he was invited to a kind of like a field trip up in Oregon, along with several other entities working to address HABs, or start working to address HABs on Lake Havasu. His region was invited, also Arizona Department of Environmental Quality and the Chimwebi tribe were invited for this meet and greet and starting to strategize on addressing HABs in Lake Havasu. And they are continuing to work on developing a monitoring plan and seeking resources to implement such a program, because where there has been, voluntary sampling, monitoring, it's well known that there is a HAB issue on in that area. There's just no routine monitoring program yet. So we're also trying to support that in ways that we can find. And I'll hand it back to Karen.
Thanks for filling in, Marissa. So now we have Region 8, Santa Ana, and we have a new HAB coordinator, Kirk. Kirk, I can't hear you, but it doesn't say you're muted. Maybe it's a headphones thing.
If you're using the browser for Zoom, in the upper right-hand corner, you might need to acknowledge using your microphone in this app.
Say something. Let's see if we can hear you.
We can't hear you,
No.
Kirk, sorry.
Maybe what if we give you a minute and we'll circle back around? Does that sound good? Okay, great. So we'll skip over back to Region 9. Carrie, do we have you on the line? I think I see her here. Carrie? Okay.
Are you, sorry, are you coming to me, Region 9?
Yep.
Okay, okay. Let me just share my screen. Oops. All right, sorry, caught me off there.
Sorry
Is
about
that
that.
working?
Looks good.
Okay. Hi, so I'm Carrie Kowalski at the San Diego Water Board, the FHAB lead, along with Debbie Woodward, and she's on the meeting as well. So brief update, looking back at 2023, here's just a little highlight of our most notable cyanobacteria blooms. They all occurred in reservoirs, and the most prevalent toxins that we saw were microcystins. So these toxins reached a warning trigger level on Diamond Valley Lake. You see there in the upper left. And the rest of the reservoirs, it reached a caution level. So that was Lake Henshaw, Lake Morena, Lower Otay, and then Lindo Lake. Last year, we also conducted a small study that focused on some of our reference condition streams where we knew that cyanobacteria were present. So that was based on visual assessments and historic data from past bioassessment work. And so what prompted this is that we often see a lot of nostoc in particular, but other cyanobacteria too at these sites. So we've been curious as to whether toxins were present in these streams. So for this study, we deployed spat for about a week, and we also collected benthic mats. And these were sites where we also conducted bioassessment last year. So we also had the algal composite from the bioassessment work, and that was analyzed for cyanotoxins. What we saw in these streams were that anatoxin was detected in the spat at all 6 sites, and one of the sites had microcystins detected in the spat, and that was Doan Creek. However, the benthic mats and the algal composites all came back non-detect for all 4 toxins. And we also found microcoleus at one of our sites. And I know Mike from Region 1 had mentioned that one in particular. Our algal taxonomist she was at CSU San Marcos was particularly interested in that culture. So we collected extra for her. She's investigating when microcoleus is toxic. So that culture was analyzed for toxin genes and toxins, and in our case, it came back non-toxic. So overall, we had good news for our reference sites. The other small study we conducted in 2023 took place in high agricultural use watershed streams that with known high nutrient concentrations. So in 2021, we deployed spat in many of these streams for some back-to-back intervals during bloom season. And then a subset of those streams were chosen to reinvestigate in 2023 in hopes of determining whether the spat concentrations we saw back in 2021 were a level of concern. And the streams that we chose are there in the center, the Temecula Creek, Murrieta Creek, and Santa Margarita River. Those are all located in Santa Margarita River watershed. So in 2023, we deployed spat again in the streams, but since it was, as everyone's been noting, a higher water year, it was later in the season. And what we did, we waited for the spat to have toxin hits to then collect mats. And in 2023, though, we didn't quite get the information that we had hoped for, probably because of the wet year. But in 2023, the toxin concentrations in the spat were much lower than what we had seen in 2021. But the good news was that all but one of the mat samples came back with non-detect for toxins. So we at least know the concentrations we saw in the spat in 2023 are not of concern. So we decided to shift gears and hopefully learn other things from this data. So we're now looking at nutrient ratios and flow data. So this study essentially is to be continued. And finally, I'll wrap up with what the San Diego Water Board has planned for 2024. We'll continue to respond to bloom reports and complaints. We will participate in the pre-holiday sampling again this year. And as funding allows, we'd like to focus this year's sampling efforts on smaller public ponds that are used for tribal and subsistence fishing, as well as for recreational uses for underserved communities. So these sites are the ones that aren't large enough for the satellite imagery to pick up and give us notifications. But we'd like to put an emphasis on the sites that have not been previously studied before through this or other programs. And that's all I have. That's the video.
Thanks so much. All right, let's go to East Bay Regional Parks District now. Hal?
There we go.
Oh, we got Sabrina.
Hi, I'm actually going to be giving the
Okay.
update today. Let
Thanks
me
for
just
doing that.
adjust my camera. Sorry. Okay. Let me share my screen. Oh, I can't share my screen, but we did send
Oh,
the
no.
PowerPoint over.
You should now. Sorry, I made you a co-host.
Oh, great. Thank you.
Thanks, Jamie.
Okay. Okay, can everybody see it in presenter mode?
Looks great.
Fantastic. Okay, well, I am Sabrina Sexton. I'm part of our water management team here at the East Bay Regional Park District. And I'm going to share some of our 2023 programmatic updates. And pictured on this slide is a really pretty bloom we had at Del Valle Reservoir this year. So getting into some of our advisories, just to give y'all a general picture. We have our green bars being no advisory, yellow being caution, and 3 being danger closure. But just to note, we do use the state's warning level as our danger level here at the Park District, just because we will close swimming, but we allow boating regardless of bloom conditions. So all of these danger markers are actually the state's warning. So this data represents the whole year, so full 365 days, even though Lake Anza and Lake Temescal only allow swimming April through October, which is the Park District's official swim season. And Big Break and Contra Loma, there is no swimming allowed ever, but they are open to boating and fishing. And then Shadow Cliffs, Del Valle, and Horseshoe Lake at Quarry Lakes are open to swimming year round. So, pretty much all of our water bodies this year were impacted by HABs blooms, and specifically microcystin toxins are what we saw the most. Most notably Lake Temescal, because it has a shorter swim season that's just April through October. It was only open for 2 weeks last year because a big chunk of those danger days were during swim season. And then Lake Anza had a pretty unique swim season this year as well, in that while we did have a few days of HABs related closure, 23 of the closure days we had were actually due to azolla fern growing out of control at the lake. So we had to close the lake due to safety concerns so that we could harvest some of that azolla, which I will get into on a future slide. We also do fish toxin testing. So we've done this every year since 2014, I believe is when it started, but 2017 is when our data sets really got consistent and we had our methods down. So in our 2023 fish tissue surveys, we collect largemouth bass from Contra Loma, Del Valle, Horseshoe Lake, Shadow Cliffs, and Lake Temescal. And the reason why we collect largemouth bass as opposed to other species is because largemouth bass are resident fish. So that means that they spend their whole life cycle within our lakes and reservoirs, as opposed to like trout and catfish, which we actually plant in our water bodies. So this is more of a study of bioaccumulation. So in 2023, we detected microcystins in most of our liver samples, but no toxins were detected in any of the mussel fillets. And we actually use the OEHA sports fish guidance, which is this chart here, table 4. So that's kind of our threshold for if we do get detections, what are our action levels. Let's see, and just as a note, I'm sure we've verbally said this on these meetings before, but we have historically had one mussel fillet detection of microcystin toxins in the years that we've been doing this, and that was in 2021. But we believe that that was due to sampler error, in that we were using the same filleting knife for the liver samples and the muscle samples and not cleaning it in between. So we have since added disinfection protocols that we follow pretty rigidly and have not had a detection in mussel fillet since. So this is kind of how we assure our friends that they can fish regardless of HABs blooms, and that the fish are safe to consume as long as they are washed and thoroughly gutted and cooked. So some of our mitigation efforts this year, we really have two main lakes that we prioritize in terms of mitigation, and one is Lake Anza and the other is Lake Temescal, just because those two lakes we hold full control over. We don't have other stakeholders involved, so it's a little bit easier for us to, craft these mitigation plans. And so, as I mentioned earlier, we had an Azolla boom at Lake Anza in 2023. So we had actually purchased an aquatic harvester at the end of 2022, I believe, with the intention of hopefully being able to use it at Lake Anza, because we also had an Azolla bloom in 2022 that we did not end up removing just because swimming was not open in 2022. So just to kind of give a rough timeline of how things happened at Anza this year. So August 15th is really when we noted that Azolla was covering the vast majority of the lake surface. So on the 16th, the swim beach was closed for safety concerns. And then on the 28th, we were able to get the aquatic harvester in there. But unfortunately, it didn't really work well for the Azolla fern removal. The buckets that we had, like the attachment buckets, were suited more towards like larger aquatic weed species. So it ended up being a combination of boom netting and raking that we were really able to remove the majority of the Azolla from the lake. Unfortunately, about a week after reopening the lake, we had a really bad HABs bloom and had to close the lake to swimming for the rest of the season. And that's pretty much that. So moving on, we also have our hypolimnetic oxygenation system running at Lake Anza. This is the 2023 marked the third year of operation for this system. And we were able to keep the hypolimnion oxygenated all year long. But our goal ratio is going to be this red line, that 5 milligrams per liter. And we did drop below that in the fall, late summer into the fall, actually. And you can see on this figure, we did actually have a HABs danger advisory that happened shortly after the oxygen was recovering. So we also do monthly watershed monitoring at Lake Anza, which we collect nutrients flowing into the lake from Wildcat Creek, as well as samples from the lake itself in the epilimnion and hypolimnion. And you can see orthophosphate is one of the key nutrients, right? Because that can dictate cyanobacterial growth. So most notably, we see that we have plenty of external and internal loading events that happen every year, and they don't seem to be correlated with our oxygen numbers in the hypolimnion. Ideally, we wouldn't have internal loading events while the hypolimnion was well oxygenated, but we tend to see that anyways. And then just to kind of give a general timeline. So in 2022, we had a huge Azolla and duckweed bloom that covered the vast majority of the year. And then in 2023, we had a shorter Azolla bloom that still covered the lake, but it was buffered on either end by caution advisories and then a danger advisory as soon as we cleared the lake. We also test for nitrate in our monthly nutrient profiles. And again, we see that we have plenty of both internal and external loading, and that these events are not necessarily tied to the amount of dissolved oxygen that we have in the hypolimnion. Again, the danger and cautions. Okay, And then Lake Temescal. So in 2023, we continued our watershed nutrient monitoring, and we also received daily remote sensor data from dissolved oxygen and temperature loggers placed in the lake as well. So early in the year, we discussed the possibility of installing a diffuser system to avoid water stagnation, particularly in the swim beach area, where we tend to get a lot of filamentous algae growth, which you can see in these pictures. It's a combination of filamentous algae and benthic algae. But unfortunately, it was deemed unfeasible due to staff availability to facilitate installation and maintain the system. So a rough timeline at that lake. We went into danger closure on June 5th, which unfortunately was just days after opening the beach. We opened the beach on June 1st. And then later in that month, we were able to do a couple of treatments. So first it was phycomycin, and then that was followed the next day with 2 Utrizorb products, one to strip the water column and the other to then bind those stripped nutrients to sediment. Unfortunately, that did not have a big impact on our bloom conditions. So on August 8th, we did a Green Clean Pro treatment, again, to try to target a lot of the filamentous algae that was growing in the swim beach area and hopefully disrupt that so that water could flow through and maybe give us better water quality conditions for the beach. So August 14th, we finally went down to a caution advisory, but that was short-lived. It only lasted about 2 weeks until we went back to a danger, which effectively ended swim season at that beach for the year. And again, at Lake Temescal, we do our nutrient monitoring. So we see a lot of internal and external loading. What is interesting is that we do see that a lot of the internal loading spikes are during times where oxygen is depleted in the hypolimnion, which, you know, this might have a stronger correlation simply because we're not artificially pumping in oxygen to this lake, but there's still plenty of external loading as well. And again, we have our HABs advisories. And here we have nitrate as well. So you can see that the pink and purple lines are all external sources. So particularly during rainfall, we saw a lot of inflow of nitrate into the lake, while internal loading compared to orthophosphate was fairly low. But, you know, these spikes did not necessarily correlate with our HABs blooms. So moving into 2024, we are going to continue treating at Lake Temescal, but we're hoping to do so earlier in the year. We've been advised by our consultants as well as a few other water quality professionals that treating earlier in the year, we might be able to prevent these blooms from getting as bad as they get. So we're aiming for February or March of this year, permitting allowing. And we're also hoping to treat at Lake Anza for the first time this year, again, permitting allowing. We plan on working on our aquatic harvester protocols because one issue that we ran into after the snafu at Lake Anza was cleaning. So we need to make sure that it's clean before it's moved from water body to water body. And then we are also concerned about special status species being disrupted during harvester activities. So we'll continue working on those protocols in 2024. And of course, we'll continue our flagship programs of nutrient monitoring, regular HABs monitoring, and our fish tissue toxin testing program. And that's it.
Thanks, Sabrina. There's a question for you in the chat, but we are running low on time, so we're going to have to move on. If you wouldn't mind responding in the chat, that would be awesome.
Yeah, definitely.
Thanks. All right.
Nice.
And let's circle back around. Kirk, did you get your sound issue worked out? Ooh, I think that's going to be a no. So sorry. Do you mind maybe sharing your slides, and maybe Marissa or Carly, do you feel like you know enough about what went on in the region to try to narrate on the fly? If not, maybe we can just send out the slides or something.
I can try if it's okay with Kirk.
Sorry to
Are they
put you
gonna
on the
back me
spot. If not, that's okay. We can just send out the slides to everybody after. Okay.
Oh,
Awesome.
it looks like he's willing.
All right, it's happening. Good luck.
I am not going to be able to do the justice of Kirk, but
I'm sure.
thank you for trying, Kirk. And yeah, so Kirk Larkin has become the new dedicated HAB coordinator for the Santa Ana region. He's taking over for Barbara Berry, who's left that position and moved on to another state agency. And Kirk has had several years of field monitoring experience in collaborating with the FHAB program. He was the lead field-based survey and investigator. So he's been part of the team and now coming on to coordinate it. And yeah, the Santa Ana region covers several counties, and there's quite a bit of activity in the Riverside County. So we'll get into that. They had many reports in their region. So in Riverside County, there's Lake Evans and Lake Paris. Lake Paris is often monitored routinely by the Department of Water Resources. Also in Riverside County is Lake Elsinore, which has a long history of oftentimes year-round cyanobacteria blooms. And also there's a concern about Canyon Lake in Riverside County, and also the San Joaquino Wildlife Area near Lake Mystic Lake as well. So Mystic Lake is actually currently still having a bloom, at least according to our recent observations. And that's on our reports map if you want to take a look at that. In Orange County, there's also some reservoirs that had documented blooms, and also some at Huntington Harbor. There are some floating mats, possibly from filamentous algae, and several lakes in San Bernardino County. One of the hotspots continues to be Big Bear Lake. Next slide. All right, so they participated in the pre-holiday sampling events and focused their efforts and the resources on Big Bear Lake in San Bernardino County, Mill Creek, and Lake Elsinore and Lake Hemet. They're pretty popular places for recreation, so it's been great to keep on monitoring there. For Big Bear Lake, they had a pretty diverse cyanobacteria, Aphanizomenon, Dolichospermum, and others. And the ELISA results stayed on the low side this year around the caution advisory. And there was a lake-wide caution advisory, and including with the sites, we could complement the field-based survey information with the satellite imagery that's shown here on the left. Next slide. All right, so for Lake Hemet, we also were able to get taxonomy. There's more microcystis here at this lake, more dominant. And there was toxin testing. Again, microcystin was the most elevated, and this exceeded the caution advisory, and that was posted again lake-wide because of its hydrology and size. All right, so additional monitoring and assessments, particularly with the partners, something that they would like to highlight. And we really appreciate all of our partners. So all of us have been trying to highlight those. So California Department of Water Resources, also Department of Fish and Wildlife, and several counties and cities that have been transitioning to taking lead on routine monitoring, including regional parks, City of Lake Elsinore, and Property Owners Association as well. And thanks everyone for hearing out Region 8's update. Thank you, Kirk, for putting together the slides.
And thank you, Marissa, for stepping in like that as well. All right, well, we're a little behind, so let's get to our break and we'll re-adjourn at 10. Let's give us a couple more minutes, let's say like 10:35, so that people have time to go to the restroom and grab some water. So we'll see you then. All right, so our next agenda item is Megan Kofer. Dr. Megan Kofer is going to give us an awesome presentation called Eyes in the Sky, Monitoring Cyanobacterial Blooms in California Waters. And she is joining us from NOAA's Center for Satellite Applications and Research. Megan, would you mind sharing your screen?
Yeah, absolutely. See how this works with presentation mode. Okay,
Looking
I think
good.
we can see just my slides now.
Yep, looks good. Thank you so much.
Okay, perfect. Yeah, thank you for having me. I'm excited to share some of the work that we've been doing for many years now, with a particular emphasis on California lakes and reservoirs. So again, my name is Megan. I'm with NOAA's Center for Satellite Applications and Research. Today, talking about cyanobacteria. This is one of my favorite photos taken from a drinking water intake, actually, of cyanobacteria that contaminated the intake and subsequently the drinking water several years ago. And here, what I really want to emphasize on this slide, potentially the most important thing I'll say the entire presentation, is that with satellite imagery, what I'll be showing today, it's not here to replace any of the field-based monitoring efforts that were talked about earlier in this meeting, and that I know are a huge part of California's efforts. But instead, they act as a complement. Satellite remote sensing, these observations are flying around the Earth almost every single day, and they can really help close some of the spatial and temporal gaps that do exist in field monitoring, and can really be used together with field observations to get a more complete picture of cyanobacterial blooms and water quality for inland systems. I think the health impacts of cyanobacterial blooms are pretty obvious, but I think we have some interesting studies as well that have looked at the satellite advantages for economic analyses. So this is a single image from a cyanobacterial bloom that occurred in Utah Lake several years ago. And in this case, the bloom occurred right around July 4th weekend. So a study that was released in 2020 looked at the avoided costs associated with the satellite imagery serving as an early warning indicator system of that bloom. So because the satellite imagery saw that a bloom might be present in the lake, field observations were then collected, and they were able to issue beach closures for the lake for July 4th weekend. And because of that, the socioeconomic benefits found by improving health incomes, human health outcomes were valued at almost $400,000 several years ago. So there can be a lot of potential avoided costs available through the use of satellite imagery for detecting blooms as well. So what I'm presenting on today is work that's been done by the Cyanobacteria Assessment Network. So this was an interagency project between NOAA, NASA, USGS, and the EPA, which began in 2015 and ended a couple years ago. And the purpose of this project was to provide national coverage of cyanobacteria concentration from 2002 to 2012 using a specific satellite called NVSat Maris. And then from 2016 to present using Sentinel-3 OLCI, which I'll talk about in just a moment. And these efforts included both scientific studies, which is most of what I'll be showing today, and the distribution of near real-time data. The Ocean and Land Color Instrument is a sensor on board the Sentinel-3 satellite. The Sentinel-3 satellite is operated by the Copernicus program, which is a European satellite. And this sensor, which we call OLCI, is on board both Sentinel-3A and Sentinel-3B. These are two functionally similar satellites that orbit approximately opposite of one another, so that they can increase the number of times they can revisit a specific location. And Sentinel-3A was launched in 2016 and Sentinel-3B was launched in 2018. And both of these sensors are still operational. They have a spatial resolution of approximately 300 meters, meaning that we can see approximately 3 soccer fields by 3 soccer fields in size for any given piece of information. And with both of these satellites being operational, we have near daily revisit frequency. Obviously, in the case of snow and ice and clouds and things like that, we're not able to measure the water surface. But in the absence of these factors, we're able to get measurements every single day from this satellite. The imagery is also collected in 21 spectral bands, and it was designed for aquatic applications. So it has a lot of the relevant wavelengths that we're interested in for looking at things such as cyanobacteria. And with each one of the satellite pixels from the sensor, we're computing the cyanobacteria index. So I know that there are several people here from SFEI, and this is the same product that's used for that. So here I'm not looking to go too much into the details of the algorithm, but I do want to talk briefly about it and how we're able to differentiate cyanobacterial blooms from other things in the water. But really what I'm aiming to do here is talk about some of the summary statistics and some of the stories we can tell with the data from the cyanobacteria index. So just briefly, what the satellite's measuring is light reflected from the sun. It interacts with things in the water, interacts with the atmosphere, interacts with things under the water, and then it's reflected, and eventually some of those rays get back to the satellite sensor. And satellite sensor is measuring the wavelengths, the strength at different wavelengths that's reflected back, and is able to use that information to measure things like cyanobacteria and its abundance within the water column. So specifically with the cyanobacteria index, this is a two-step algorithm. So here we're just showing a spectral curve for the remote sensing reflectance, with the green line representing cyanobacterial bloom and the blue line representing no bloom or relatively clear waters. And we're zooming in on a specific section of the spectra here, where we first use reflectances at 665 to 709 nanometers to determine cyanobacteria presence. And these spectral bands are capturing the spectral properties of chlorophyll. So that's how we're able to take step one of the algorithm and look at chlorophyll within the water column. And then to differentiate that from other blooms that also have chlorophyll, we use spectral wavelengths spanning 620 to 681 nanometers as an additional step, serving as exclusion criterion, because these spectral bands capture the spectral characteristics of phycocyanin. So that's how we're able to take the spectral measurements measured by the sensor and determine the approximate abundance of cyanobacteria in the water. So with that being said, here, what I'm planning to show is using the Sentinel-3 satellite series, look at the occurrence, the spatial extent, and the temporal frequency of cyanobacterial blooms for the state of California for the year 2023 at both the state scale and at the regional scale for each of California's 10 hydrologic regions. So for each one of these three metrics, occurrence, spatial extent, and temporal frequency, I'll talk about the specific methods for those. But just as a general introduction, each one of these questions came at the request of stakeholders throughout the process of the CYAN project, and they were answering key questions that stakeholders were asking about blooms in their area. The first metric, occurrence, answers the question of how many lakes are impacted by cyanobacterial blooms. The second metric, which is spatial extent, answers the question, how much of the water's surface do these cyanobacterial blooms cover? And the third metric, temporal frequency, answers the question, how often do these cyanobacterial blooms happen? So across the state of California, given that Sentinel-3 ULCI has 300-meter spatial resolution, we have 88 lakes across the state that we can monitor. There are certainly some lakes that are technically able to accommodate a 300-meter pixel, but we do have additional exclusion criterion that we add to the lakes that occur across the state to make sure that we're getting the highest quality information that we can. So these include things like making sure we're not too close to the shorelines, and things like that, so that we're not getting any interference from the shore or from nearby land. So with our exclusion criterion that we defined for this project, we have 88 lakes that we can monitor. And then this map is just showing the 10 hydrologic regions across the state and how many lakes are in each one of those. So you can see the region with the highest number of lakes is Sacramento River, which is located in the northern half and has a lot of the lakes within the valley. And then we have a couple regions that only have 2 resolvable lakes that we're assessing. So the cyanobacteria occurrence, so this is just a conceptual diagram that walks through how we're computing this for each month across our entire year of 2023 for this time series. So again, this metric is quantifying the percentage of lakes across an area that are experiencing a bloom each month. So in the first step on the far left, you can see our resolvable lake with the satellite pixels within that lake. And then in the second column, you can see Each one of these pixels classified as either bloom, no bloom, or no data. So this is from the CCHAB algorithm, whether or not we're meeting the sensitivity threshold of the sensor to determine that there is a bloom present within that satellite pixel. So for the purposes of this algorithm, or for this metric, we said that at least 10% of the pixels across the lake have to have a cyanobacterial bloom for us to categorize this lake as experiencing a bloom. So in this case, this lake did have at least 10% of its pixels shaded as green. And then in the third step, we can just look across an entire area, whether that be a region or the state. And we can count how many blooms were classified as, or sorry, how many lakes were classified as bloom versus no bloom across the area. So when we do this across the 88 lakes for the state of California, we find that occurrence had a maximum value in late summer and early fall, which is consistent with previously published ecologic patterns and what we would expect. So this can also serve just as an additional source of validation that the satellite imagery is producing expected results across the state. And it reached a peak of 33%. So that means that at its maximum for the year 2023, at least a third of lakes across the state were experiencing cyanobacterial blooms. And the minimum value occurred in late winter, early spring, reaching the lowest value of 7%. So that tells us that also across the entire year, approximately the lowest number of lakes that we're seeing blooming is about 1 in 10. So again, this is also consistent with previously published ecological patterns and serves as additional support of the satellite algorithm. In addition to looking at statewide results, we can also look at regional results. So again, we see that the highest values tend to occur in late summer and early fall, and the lowest values tended to occur in the winter. And looking at the regional results can help us tease apart some of the differences across the state. We found that the San Francisco Bay Area experienced a large late Susan bloom, which you can see by that brown line that peaks kind of in November and December. And that for the other regions, patterns were pretty consistent, and there wasn't a ton of variability within regions for what we're seeing across the year and from region to region. So then we can move on to the second metric, which is spatial extent. So again, this quantifies the percentage of satellite resolvable lake surface area experiencing a bloom each month. So with this metric, again, we've got a conceptual diagram here that shows the satellite pixels for the lake shaded as either green, blue, or white, representing either bloom, no bloom, or no data. And then we can just simply compute how many of those pixels are shaded as green compared to how many pixels there are across the entire lake. So in this example, we had 4 divided by 10 or 40%. So for this conceptual diagram, our lake scale spatial extent was 40%. And so expanding this conceptual diagram to an entire region or entire state, we follow the same exact methods and are able to provide larger scale measures of cyanobacterial extent on a monthly basis. So for the entire state of California, we saw that cyanobacterial blooms covered the largest area again during late summer and early fall, pretty consistent with the occurrence metrics that we saw for the entire state. And it reached a maximum of 16%. So again, at its peak, 16% of resolvable waters across the state of California had cyanobacteria present in the water column. And we reached our minimum value 1% during late winter. So it gets pretty close to no blooms across the state at its lowest throughout the year. With this metric, the regional results were a little bit more interesting in that we saw a lot of regional variability. So we can see from those kind of peak lines throughout the year that there's different locations that are experiencing their highest spatial extent throughout the year. Some of the regions had higher values earlier in the year, and then some of them followed more of the national scale pattern. And then also what was pretty interesting here is that the South Coast had pretty consistently high spatial extent compared to the other regions. And conversely, North and South Lahontan, I hope I'm pronouncing that correctly, had consistently low values throughout the entire year. So again, some of these metrics looking at the regional scale can help tease apart some of the differences we're seeing across the state. And then finally, the last metric I'll present on today is the temporal frequency. And this is computed a little bit differently than the others in that instead of getting values per month, we're actually getting a single value per year, but we're getting that value for every single pixel that we're monitoring across the state. So this is looking, if you can imagine from the image on the left, that we have the 52 weekly composites that we're computing with the CI-cyano index across the entire year. And again, we're just looking at the percentage of those pixels across time that indicate that a bloom is present. And this tells us how many weeks across the year that a specific satellite pixel is experiencing a bloom. And then once we get these pixel scale results, as you can see on the right, we can average them across a lake, a region, or a state. So we'll show those results as well at the regional level. But first, I wanted to look at just a couple examples. So I know Lake Elsinore was mentioned earlier this morning, and yes, it has cyanobacterial issues. And again, we can see that with the satellite imagery. So this shows the cyanobacterial frequency measures, and from the scale on the left, you can see that yellow values are the absolute highest from the satellite imagery. And here we're getting values at or near 100% for the entire year of 2023. And I do want to add that satellite imagery is not detecting any toxins. So it's not something that we can measure with the data. And also, just based on how the metric was computed for this instance, it's also not giving us a measure of the strength of a bloom. So it's just saying that a bloom is present, something above the detection limit of the sensor, which is difficult to quantify, but we currently think it's between about 10 and 20,000 cells per milliliter. So based on that, the entire lake for the entire year was experiencing cyanobacteria in concentrations above the detection limit of the sensor. I know that Lake Elsinore is well known to have cyanobacterial problems, but this is also just a comment from Lake Watch across the lake, where caution advisories were issued across the monitoring sites for the lake across the year 2023. And then another example, Eagle Lake, located in Northern California, showing the cyanobacterial frequency for the year. So again, the scale goes from dark blue being the lowest frequency up to yellow being the highest frequency. And with this example, we can see a lot more variability across the lake. So these pixel scale results can reveal spatial patterns. So for instance, we can see areas where cyanobacterial blooms tend to occur, and help us highlight areas that are more prone to blooms. And then they can also help prioritize sampling efforts. So if we had no other information about a lake, we could use results such as this to try to target where across the lake we want to go collect field samples. So looking at areas with higher frequency might help us be able to collect information about toxins within the water body. And then they can also help put point-based samples into the context of larger ecological patterns. So again, for instance, if we had just a single point observation that tended to be measured for this lake, if it's located in an area that the satellite imagery suggests has high cyanobacterial frequency, we can think that maybe that point-based sample isn't representative of the entire lake, and that conditions elsewhere might actually be better than where the field samples are tend to be collected for that area. And then, as I mentioned, we can average these across an entire region. So we're just taking those pixel-based results that I showed for Lake Elsinore and Eagle Lake, and we're averaging them across an entire region. And so each one of these donut plots is just essentially a pie chart showing the percentage of cyanobacterial frequency. So again, kind of the percentage of time that cyanobacterial blooms tended to occur. And so here we can see that the highest area with the highest frequency was the south coast, where Lake Elsinore is located. So very likely that Lake Elsinore is playing a large role in that high temporal frequency for the region. And then we can also highlight regions like the Colorado River, where cyanobacterial blooms tend to be less problematic and occur less frequently. So to go back to our three metrics and just talk quickly about the findings for each one of those across the state. With the occurrence metric, we found that results were highest in the summer and lowest in the winter, which again is consistent with previously published ecological patterns. But with the full year-long satellite imagery that we're able to get from the Sentinel-3 satellite and some other areas across the country, we're actually finding that cyanobacterial blooms are really prolific throughout the winter months. So it's interesting to be able to look at these on a regional basis as well. And then at any given time for the year 2023, at least 7% of lakes had cyanobacterial blooms. With the spatial extent metric, We also found that results indicated the highest extent of cyanobacterial blooms in the summer and the lowest values in the winter. But with this metric, we saw a lot more regional variability. So those blooms are covering a lot different areas of the resolvable water area from region to region. And then with the temporal frequency measure, this can be really useful for prioritizing sampling efforts, and then also can reveal lakes that are experiencing chronic blooms throughout time. So with the cyan analysis, as I mentioned, about 88 lakes across the state with the 300-meter Sentinel-3 data. But I want to talk a little bit as well about where we're going next with the, we haven't fully figured out the name yet, but the Cyanobacteria Assessment Network 2.0 or something of that effect. Cyan 2.0 is what we're calling it for now, which just began last year. And this is an interagency project using the original agencies and the Cyan project, which included NOAA, NASA, USGS, and EPA. But additionally, we're collaborating with the U.S. Army Corps to get additional feedback and assistance throughout our efforts. And the idea of this project is to provide, again, national coverage of this time chlorophyll A concentration from 2015 to present. And again, our efforts will include both scientific studies and the distribution of near real-time data. So with these efforts, we are using another European satellite called Sentinel-2. And again, Sentinel-2 has both Sentinel-2A and 2B, which are functionally similar satellites positioned 180 degrees offset of one another to increase the temporal frequency. And each one of these has the multispectral instrument, or MSI, on board. And Sentinel-2A was launched in 2015, and then 2 years later, Sentinel-2B was launched. And again, both of these are still operational. And with this, we have much finer spatial resolution. So we're able to get down to between 10 and 60 meters, depending on the spectral band. But the temporal resolution is poorer. So here, instead of daily revisits, we're getting imagery only about every 5 days. And that's with the second sensor included for the years 2015 to 2017. That revisit frequency would double to 10 days. Here, the imagery is collected in 13 spectral bands, so less spectral bands than Sentinel-3's ULTRA sensor, and also not specifically designed for water. So we don't have the same spectral resolution to be able to quantify cyanobacteria, and instead we have this broader product of chlorophyll A. So I know I talked through these a little bit, but I just wanted to kind of highlight those differences between Sentinel-3 and Sentinel-2. So again, and the bolded text is kind of what's preferred. So on the left with Sentinel-3, we have a much improved nearly daily revisit period. And we can use it to estimate both chlorophyll A and cyanobacteria concentration at the surface of the water. But with Sentinel-2, much less frequent revisit period of 5 days, and can only be used to estimate chlorophyll A. But with Sentinel-2, we're able to observe nearly 400,000 lakes, reservoirs, estuaries, and rivers. So we're able to get into much smaller systems. We're able to get more detailed information within the lake and also closer to the shoreline. And then also we can get into additional systems, including rivers, with this higher resolution data, compared to about 2,000 lakes and reservoirs across the US that we were able to monitor with the Sentinel-3 sensor. So again, this is a map of California with each of the 10 hydrologic regions. And I'm showing now instead the resolvable systems using the Sentinel-2 project. So here we have nearly 16,000 systems across the state that we'll be able to monitor. And broken up by each region, we again see regional patterns on higher and lower areas. But again, the coverage is just incredible compared to what we were able to get with Sentinel-3. So we're really excited to see where this continues to go in the next few years. And an example from near the road, down the road near where I live, this is Jordan Lake in North Carolina. And on the left is an image from the Sentinel-3 sensor with 300-meter pixel size. And on the right is the same lake, same day, but with 20-meter Sentinel-2 imagery. So you can see that, again, not only within the lake are we able to get much higher variability and detail for each one of the pixels, we're also able to get a lot closer to the shoreline and within a lot of the more narrow reaches of the lake. So just a lot more detailed information that's possible with this sensor. And then on the far right is a single 300-meter Sentinel-3 pixel shown with the larger red square. And then within that, we have much finer Sentinel-2 pixels at 20 meters. So it really helps us understand kind of the increase in scale within a pixel, in addition to the additional areas across a lake or reservoir that we're able to monitor with finer detailed imagery. And with each one of the agencies, we all are kind of playing a different role in this and are in year one currently. So as part of our year one efforts, we were first able to identify areas of interest. So we've chosen 4 states across the country that we're focusing our initial algorithm development efforts on, which includes Oregon, Ohio, Florida, and California. And so this is one of our test states that we're working on going forward. And as part of our year one milestones as well, we scraped the literature for all Sentinel-2 chlorophyll algorithms available and are currently in the process of creating a scoring rubric to decide which algorithms we want to use. And then once we subset the list of all algorithms down, the next step is to start testing these against satellite reflectance values for each one of our 4 test states. Which will help us understand if there is the ability to do kind of blanket algorithms across the entire country via these 4 test states, or if it will be something that's a little bit more localized, whether that be for different land cover types, different climate regions, or things like that across this country. And then also facilitating user engagement. So in each one of our states, we do have partners that we're working with from a field data perspective. And also, I mean, we cannot be everywhere at once. And so having local experts for each one of these areas is incredibly valuable for us to be able to interpret our results, understand where we're doing well and where we're struggling in our algorithms. And then moving forward in years 2 through 5, finalizing our algorithms and processing, and then we plan to have both beta and final operational release for near real-time delivery of this chlorophyll product. And then again, the metrics that I showed here for the state of California, we've computed those with Sentinel-3 for the entire Maris and Olchi time series. And we want to recompute those using Sentinel-2 data, which will be really interesting to see how the patterns that we were able to tease out with Sentinel-3 hold for the Sentinel-2 data. And then here I've linked QR codes for the manuscripts accompanying the cyanobacteria occurrence, spatial extent, and temporal frequency studies. And feel free to send me an email with each one of those manuscripts. We have the code, it's all open source in R, to be able to download the CI Cyano data products from NASA and process them into the algorithms, into the metrics that I shared today. So if there's ever a need for assistance implementing these metrics or getting the code running for your area, please feel free to send me an email. And then I've also been working really closely with Jamie Smith and Ali Lee on California specific efforts. So they've done an amazing job of not only recreating what I've done for California, but looking at more than just those 88 resolvable systems with Sentinel-3 and trying to create California specific data sets, resolvable lakes that are specific to the state, and also updating some of the efforts because there are several lakes that are now neighborhoods across the country. So yeah, changes like that, they've done a really great job on getting that rolling so that it can be a really California specific product. And with that, I'm happy to take any questions.
Thank you so much. If anyone has questions, they can just unmute and go ahead and ask. All right. Well, if you think of questions in the future, feel free to drop them in the chat and Megan, maybe you can answer there. For now, thank you so much. I really appreciate your presentation. Super interesting. Yeah, so next we're going to have an introduction from our new US EPA coordinators at Region 9. Many of you know probably Sue Keidel retired in October, late October, early November. And so we have a new team taking her place. And so, Ina, if you wouldn't mind sharing. Awesome. Thank you so much.
Okay.
Looks good.
All right. Well, thanks for having us. Hi, everyone. We have a few folks here today from EPA Region 9, and in case there's anyone newer, in addition to California, we also serve Arizona, Nevada, Hawaii, Pacific Islands, and 148 tribal nations. And so I'm Yana Kwok. I'm the sort of point hub coordinator, but there is a hub team, and we have two out of several other folks that are going to be presenting with me today. So I'll just let them introduce themselves.
Hi, thanks for having us. My name is Dana Michaels. I'm also at EPA Region 9 in the Water Division with Yana, and I'm with our San Francisco Bay Program Office.
Hi, I'm with Air and Radiation Division in the Community Programs section, and I'm Eva now.
Great. So the goal for today's okay, getting things figured out on my side. But yeah, the goal for today's presentation, in addition to introducing some of our newer faces, is also to just provide an update on some of the work that we've been working on around HABs in Region 9, in our Region 9, and which also, so that'll include, clarifying what our roles are in the agency and, informing that on our authorities. And we'll provide an overview of some of the work that our HAB team is focused on right now, spotlight some specific EPA programs that are relevant to HAB work, and discuss research and lab support and just ways to stay connected with us. For starters, EPA, and maybe this is just super basic, but EPA is the lead among federal agencies for freshwater HABs, and NOAA is the lead for marine HABs. And each agency, we have our own set of legal authorities that define what we can do. And so for EPA, we have three major authorities that I know for our team here, it was super helpful to lay it out really explicitly in this way. So I hope it's helpful for you as well. But under the Safe Drinking Water Act, EPA, we have we're given direct authorities for public water systems, but not private wells. In EPA, we tend to play an oversight role for state drinking water programs, and then can do more direct implementation for tribal systems, and can and have published drinking water health advisories. With the Clean Water Act, EPA, we can do a whole slew of things, but that I'm sure a lot of you are familiar with, related to issuing permits to reduce the nutrient loads, reviewing, approving water quality standards, and recommending criteria, and providing assistance for listing impaired waters and developing TMDLs. And both statutes authorize EPA to award a lot of various kinds of federal assistance funds that I'm gonna provide talk about more later. But when it comes to HAB response, it's primarily the state or local agencies that have the authority to respond directly to HAB events. CERCLA allows EPA to respond to HABs under specific circumstances, such as during a public health or environmental emergency, and if no other person with the authority and capability is able to respond in a timely manner. So given this, I know I've spoken with some of you before about how we initially were thinking of working on like a EPA Region 9 response plan, but once we recognize that we have rather limited HAB response authorities, we shifted our focus to be more about developing a more internal facing communication plan, which is still in development, but we hope to be at a good place for this next peak bloom season of 2024. But the goal of this is to really establish protocols within our region on how to communicate and coordinate across programs when there are major blooms, when when we're fielding different inquiries that come from the public or from you all, and just equip our all of us as staff and our managers on how to answer the question, what can EPA do about HABs more easily? So in addition, like in tandem with the communication plan, that's the growth of a work group. Actually, I'll speak more to that in a second, but some other things that we're working on, since there's a lot of new hires in the past 2 years, we've been focusing on just building relationships, internally and with external partners, and tracking needs to identify areas where EPA can be of assistance. And several of us sit on different work groups, provide input, and attend or help facilitate different HAB-related meetings in our region. In addition, we support research that is done by our agency's Office of Research and Development. So, as I introduced myself earlier, I'm the HAB coordinator, which in some ways make me the Sue Keidel successor, but really before she retired, she gave us a great gift of helping set up a HAB work group. And this is kind of a diagram of like which programs and who the people are that are involved in this work group as of today. And my role as a coordinator really is to help coordinate, help a lot in the coordination and planning amongst this group of staff that's listed here. And it's been really great. We meet weekly, we check in on HAB incidences in all of the, in our region that are reported. And then we report out on all the different meetings and work groups we're sitting on and convene around HAB concerns that come up in our respective programs, and just make sure our staff and management we're all made aware of things that are relevant to each other's work, because HABs is very much touches on a lot of our programs. And I think together we've been really growing a lot just institutionally, just better clarity on how all of our programs work together and how we can also communicate that better, how our programs work together to address HABs. So, on that note, some of this maybe might be very familiar, especially if you're someone who works for the state and works with these EPA funding programs often. But I thought it'd be really helpful to maybe frame a bunch of EPA resources through our Clean Water Act and our Safe Drinking Water Act. And this is not a comprehensive list, but these are all links that hopefully I think can get shared out after this meeting and are good starting points to learn more. But my hope today is to really spotlight a few of these programs and resources to paint a bigger picture of how these big programs really work together and can fund different stages such as planning or monitoring and project implementation. So I'm just going to start off on the planning side of things. One of the grants programs that I manage, not for California, that's a different project officer, but so that you are aware, 604B is for water quality management planning. It's a grant to states only, this one, for planning activities, and they cannot be used for implementation of control measures such as installing BMPs. And each state uses these funds differently, but water quality planning activities can be interpreted fairly broadly and can include, but it's not limited to, work such as ambient monitoring, developing water quality standards or TMDLs, or developing, watershed or water quality management plans. And then 106, and now there's some distinctions in how this program plays out for tribes versus states, but this still provides the big picture around how Section 106, it authorized EPA to provide financial assistance to states and tribes to establish and administer programs for prevention, reduction, and elimination of water pollution. And some of the it's mostly known for water quality monitoring, but I thought it'd be interesting to note that it also can be used for outreach and education, as well as training and travel. Oh, that's an important part, including HABs. So NSTEPS, if you're not familiar already, stands for Nutrient Scientific Technical Exchange Partnership and Support, NSTEPS for short. This is an agency-wide program that provides you all with technical assistance and connects you with an expert who has experience developing water quality criteria. And they pair you with staff to help you with all levels of water quality standard development. I know a lot of groups, don't have resources to work on standards from start to finish. So this is a service that EPA offers, and some tangible examples of how this could be helped or used includes like it could include like translating narrative criteria related to HABs and nutrients into numeric criteria that can be used for monitoring and assessment, or even developing more site-specific criteria tailored to local conditions, uses, and data. On the project implementation side of things, I want to highlight two programs. The 319 non-point source program is for implementation of watershed projects and non-point source strategies to tribes, territories, and states. And eligible activities include restoration and protection projects like riparian restoration or plantings, exclusion fencing, and for different projects related to stormwater runoff or source water protection. And for the other project, the implementation program, I'm going to let Dana speak to the SFA program.
So the last Clean Water Act program that we wanted to highlight for you is the SFA program office at Region 9. And this program includes the San Francisco Bay Water Quality Improvement Fund, which is a competitive grant program that has been running since 2008 with a focus on improving water quality to the Bay itself. So the geographic scope is the 9 Bay Area counties that drain to the Bay. So in order for your project or work to be eligible, that's the geographic scope. Project categories include restoring wetlands, restoring water quality, greening development or stormwater improvements, and a community capacity building EJ category. So in this blue slice of the pie, you can see how much of our funding has gone towards restoring water quality projects, which is really where the HAB-related and nutrient-related work that we have funded in the past has fallen. So that work that has been eligible and funded in the past includes some nutrient reduction pilot projects at wastewater treatment plants around the Bay, and also science programs to build an understanding of nutrient cycling and HABs in the Bay. So those are just examples of projects that have been funded in the past through the program, and other sorts of HABs or nutrient-related work could be eligible in the future. Additionally, in December of 2022, Congress authorized the San Francisco Program Office to expand and become a new EPA program office. So this is really exciting, and with this authorization comes increases in funding and additional responsibilities, including the development of an annual priority list. The first iteration of this list includes nutrient management as one of the priorities, and so the program office will be able to fund work that supports that priority in the future. And Luisa Valiela is the program lead for the SFA office, and her contact information is there, as well as a link to the SFA Water Quality Improvement Fund site in case you want to look further into it.
Great. And now I'm going to transition and spotlight two programs authorized through the Safe Drinking Water Act and the Source Water Protection Program. And yes, this does kind of have some overlap with the 319 non-point source program, because that is also eligible for source water protection projects. But this one it's a voluntary program, and it's intended to protect both underground and surface sources of drinking water. And the premise of it is to encourage states to develop and implement a source water protection program. And so EPA provides funding through a set aside of the Drinking Water State Revolving Fund to conduct source water assessments and implement projects for federally regulated water systems. And I just share this to flag that it is a resources that EPA offers to the states. And there's some really good case studies from other states, such as Iowa, where they've used this program to fund agricultural BMPs to help manage nutrients with the intent of preventing HABs. There are also further links throughout this presentation for a lot more information on just other general resources too, and education, best practices, and different technical assistance that's available for assessing and protecting source water. The other drinking water program I really wanted to mention today is that the tribes, there are these 2 new grant programs to address emerging contaminants that tribes can apply for, that can, fund, cyanotoxins are considered emerging contaminants. So, one of these programs is the tribe Drinking Water Tribal Set Aside Emerging Contaminants Program. That's a bit of a word soup there. And the other is the Emerging Contaminants in Small or Disadvantaged Communities Grant. And these 2 grant programs, well, they normally address, they emphasize a little bit more the PFAS work in drinking water. Projects that address cyanotoxins are eligible. And I really want to spotlight this particular set of programs because the funding is still open on a rolling basis. And combined, there's a total of around like $14 million in potential funding to address emerging contaminants in drinking water. And there's no cost share requirement or no underserved community requirement. So I was asked to make sure I emphasize this opportunity to this group and to share the word. And so there's a QR code for more specifics on the Region 9 program on this. I'm gonna skip along for time and allow Dana and Irina to take over to talk about some of our research.
Thanks, Yana. So in Sue's retirement and absence, I've been taking over coordinating one of our Office of Research and Development, which I'll say ORD from now on, research projects. And so I'm going to speak a little bit to the ORD research and work going on along with Irina. So ORD's aim, in addition to their mission, is to provide the scientific basis for the policy and decisions developed by our program offices. And so we're going to spend some time talking about a few of the studies that may be of interest to this group, but there really is a wide array of research happening on HABs and many more subjects within ORD, but just with limited time and for this audience, we'll just speak to a few. So next slide, Yana. 2 projects we wanted to highlight include the top figure here in the first bullet on work underway to explore early detection of cyanotoxins by qPCR and RT-qPCR. So the top figure is showing a case study from Smithville Lake in Missouri, where the microcystin gene MCYE, DNA and RNA was detected before, shown by the first arrow pointing at the green dot, on the order of magnitude of weeks before the microcystin toxins were detected by ELISA, shown in blue, the second arrow to the left. So that is work conducted by Jinrang Liu, and his contact information is there if you are interested in reaching out to him. There's also been other case studies, including one that explored the same approach with anatoxin genes in a lake in Washington state that showed the detection of RNA 1 to 3 weeks before toxins were detected by ELISA. So this is really exciting potential for cyanotoxin encoding genes to enable short-term predictions of cyanotoxin production. The second bullet point and map lower down on this slide is a project on modeling the risk of toxic HABs across lakes in the US. So this project combines satellite imagery from Cyan, which you just heard about in the past presentation, along with field samples from the National Lake Assessment to model risk among all these lakes. So this modeling effort identified U.S. lakes that were at high risk for developing toxic HABs. And the whole premise of this was to allow for additional monitoring to be suggested or risk communication at these lakes. And the lead researcher for this project is Amalia Handler, and her contact information is there as well. And then lastly, the One Environment, One Health. We wanted to highlight this research webinar series that EPA ORD is hosting. The first of the series is actually happening concurrently with this meeting, but I'm sure it's being recorded or you'll be able to catch the next one in the series. But the focus of this is to communicate the latest research related to nutrients and priority impacts of nutrient pollution, so harmful algal blooms and hypoxia. And so the webinars are going to be held every other month from 2 to 3 p.m. Eastern time on the last Wednesday. So there'll be more information on that in one of the later slides. Next slide. And then the last project I wanted to share about with regard to ORD, some of you on this call and many of our amazing partners in California are closely involved in and aware of is the regional ORD Applied Research Project. So you may hear this term ROAR, and that's what it stands for, on developing standardized methods for sampling, analyzing, and assessing benthic HABs. So this effort is conducting pilot studies in 7 of EPA's regions across the US at streams and rivers that have recently experienced benthic HCBs. And there are pilot sites in California, including the South Fork Eel River and the American River. The analyses are including toxin analysis, cyanobacterial composition, areal extent, and thickness. And the study is aiming to benefit tribes, states, and other federal agencies by providing a comparison of methods that crosswalk both optimal sampling protocols and analytical methods. And we just want to take a moment to give a huge thank you to our California state partners on this project in Region 9. This project really would not be possible or successful without you. And the folks involved in this project have been critical at every step of the way. So from the formulating of this idea and sharing concerns about benthic HABs, sharing expertise, hosting the training, and actually carrying out the field sampling. So we just really want to give a huge thank you to all of the folks that have been involved in this effort. And I'll pass it on to Irena to speak to her research.
Thank you, Dana. So EPA Region 9 Air and Radiation Division is mostly involved right now with HABs in terms of supporting and assisting ORD and looking at wildfire deposition and its impact on downwind HABs. So we're working from the data set looking at a 15-year period of PM2.5 data, including smoke plumes, to identify those nutrients that are elevated in air samples and on smoke impacted days to look for possible correlation. So right now, our goal is really to utilize the remote sensing data available to assess this relationship. Air Division also wants to improve and assist in research on aerosolization of cyanobacteria in the future. And we plan on participating more in the communication plan for possible wildfire and HAB events.
All right. So yeah, in addition to supporting research, we within our laboratory services and applied science division, we have our Region 9 lab who has been providing free lab analysis for a variety of external partners since early 2000s. And they've been providing analysis for about around 800 samples a year, and the analysis that they provide is only so far for microcystin using the ELISA method, and there is ongoing and continued support for the Klamath Tribal Water Quality Consortium, and The lab, though, is really intent on looking to expand its capacity and capabilities for like not only additional samples, but also additional toxins, and also drinking water methods in the future. So, I hope to keep in close touch to you all whenever that, can happen. So, but I will say the best way to, for both science and lab support needs, and in general, honestly, you can connect with me as the hub coordinator. And what I do is I keep track of the different requests, or we make note of the different science or lab support needs and interests. I communicate that to our regional science liaison and our lab manager, and ultimately they sort of have a final say in what takes priority and what they have capacity for. But the best way I can communicate and advocate for you all is if I'm well aware of the different needs, interests, and priorities that you all have too. So I'm just really looking forward to just continuing getting to know a lot of you on this call. And yeah, this is our final slide. There are just a couple other ways to stay connected. Well, the first one, the Sinus Symposium, this already happened in the fall, but I think headquarters especially has been very proactive in trying to understand what the different education and outreach and webinar and training type needs are. And so this was a response to help equip especially newer scientists. Our target audience for the symposium were more beginner, novice with HAB science, and we really successfully reached that target based on like surveys of who attended, and got some really good insights on future topics to host webinars around. And I also just wanted to give a shout out to Shannon Murphy, Thomas Hayashi, and Karen Atkins for presenting on the fourth day for the California Spotlight. So thank you for your participation and your sharing your great work for others to learn. A lot of people really look forward to hearing about California's work. In addition to that, there's a link here from the same ORD webinar series that Dana had mentioned. And one thing I really want to flag for you all is that in the fall, that EPA and I forget all the other federal agencies that are involved in the planning of this, but there will be a HAB symposium that will take place in Portland, Maine, October 27th to November 1st. It's aimed to address the different challenges, and it's supposed to be a good opportunity to engage in a lot of meaningful discussion and explore different innovative solutions. Headquarters asked us to do this plug about a logo design contest, if that's of interest to you, or if you have knack for art, or kids who have a knack for science and art. There are deadlines for the logo design contest for this national symposium that's due on February 6th. So with that, I'll just stop sharing, and I think we have a few more minutes if there are any questions. I haven't had a chance to look at the chat yet.
Yeah, thank you so much. This is just so many resources. Very exciting to hear about everything you're doing. It looks like Sarah Ryan did have a question for you. Her question is, are 604B funds open to tribes?
Oh yeah, so 604B funds, it's not it's not necessarily directly fundable to tribes, but there is but if states are working on like a water quality management planning project in partnership with tribes, and or if it happens to benefit tribes, like that, that's totally like an eligible use of the funds and the planning work too. So I hope that helps. So I think it's more important that you get in touch with the California 604B program manager for specific questions, because each state really uses the funds a little differently.
Thanks. Does anyone else have a question? Feel free to unmute and ask at this point.
This is Bill Taylor.
Yep.
Can you hear me?
Yes, we can.
I thought the study on benthic HABs in the streams is great. My question though is, did you do a similar thing in lakes? I know in my experience, we found benthic HABs pretty regularly in at least one of our lakes, mainly on the big rocks on the face of the dams. They loved it.
Yeah, thanks for the question and comment. This study is focusing on streams and rivers, just because the methods that we're testing, it's one of the variables that they're trying to constrain. But yeah, certainly I think there's going to be a talk later today in the lineup that may have some other thoughts about, expanding benthic work to lakes, but at this point that the scope of the project is streams and rivers. But yeah, it's a hope for the future that we can keep expanding benthic studies to other environments where they occur.
Thanks, Bill. Anyone else? All right, well, you can always drop a question in the chat. Thank you so much for your presentation. I really appreciate you all taking your time to talk to us. It's so informative. And now the final kind of agenda items before lunch here. We're going to go through our CCHAB subcommittee updates, of which there will be 3 kind of end of the year discussions, starting with Dr. Shannon Murphy, who's going to talk about our illness work group.
Great. Thanks so much, Karen. Is it okay if I just share one slide?
Of
Can
course.
I go ahead and do that? Okay. It says I'm disabled.
Oh, Jamie, would you help out with that?
Great.
Thanks.
Okay.
All right, try it again. Sorry, I thought I'd added.
All right.
Already.
Hopefully you folks can see that. It should be a green screen, if you will. So this is just a quick overview. So for those of you who are quite familiar and those who may not be so familiar with our efforts, do you see it? Okay. So California has the HAB-related illness tracking work group, and it's comprised of 4 state agencies. So the 2 health agencies, one that I represent, OEHA, it's a subdivision of Cal EPA, and then we have the California Department of Public Health, and then we have, of course, our team with the Water Board, both the state and the regional boards, and then the California Department of Fish and Wildlife. And so we work together to investigate potentially HAB-related illnesses in humans, domestic animals, so especially dogs, and then livestock in a few cases, and then wildlife. And so this is just a real quick overview flow chart of what our efforts entail. So we get our report. It could be through the web portal, which is most common, a California poison control case, or direct report from partner agencies. Those could be park managers, FHAB coordinators, and the like. We conduct the interview. The agency representative that takes lead is often driven by the type of case that it is. So in the humans, it's CDPH with sometimes OEHA, and then OEHA usually does domestic animals, and then Karen and company take over the wildlife cases. Then we, if it's appropriate, do site visits and coordination. So that could be observations, working either directly with our HAB coordinators or other agency partners, looking to get environmental data, collecting satellite data, but then also going out and visiting the site to get water and algae samples. CDFW might do a site visit if it involves wildlife response, and then of course lab submittals, and then other partnerships as it's appropriate. And then once we get that going, that's part of our investigation. It's a multi-agency coordination effort. And then it's really critical in the first couple of days of getting a report to be able to interview those reporting parties. So as was mentioned earlier, when we get these reports in, having contact information where we can directly contact the people on the ground, whether it be the responding agency or the member of the public or tribal leadership that is reporting the incident. So being able to get those first-hand narratives is critical. And then we can combine all of these efforts together to make our case. And then we have a meeting every 2 weeks at least, and then several interim meetings or email correspondence with the 4 agencies as needed. And we come up to a case determination as a consensus. So all 4 agencies have equal participation in the discussion, and we make our determinations. And really it falls into 1 of 2 categories. Is it a HAB-related illness or death? Or is it not? And in some cases, unfortunately, we just don't get enough evidence to say that it is HAB related. So it could be the case where there might be likelihood of it being HAB related, but we don't have either the environmental evidence, we can't get a hold of the reporting party, or there's just some key elements that are missing that don't meet that burden of proof under the CDC definitions for it to be a HAB related case. Those that are determined to be HAB related are reported not only for California tracking purposes at the state level, but also to the One Health harmful algal bloom system for the CDC. And so ultimately that goes into our efforts. Currently, the HAB illness work group is reviewing our final numbers and getting our case reports into OHABs for 2023. We should have those numbers for you at next meeting. We didn't want to do it prematurely here. We want to make sure that everything's good and that there's no discrepancies. Also, too, we want to do some trending analysis now that this will be our 6th complete year of data since we've been tracking these illness cases. So we'll be able to show you some differences. Does it go down this year, up? Was it more represented in human cases versus wildlife and things of that nature? So I think it would be helpful to have some of those kind of overhead takeaways next time. A few other things since we last met in October. The HAB illness work group has confirmed with CDC OHAB's team that we have reconciled and gotten rid of any questions regarding our 2022 cases. And that's really to help double check us as a state, but also to make sure that CDC is shored up to produce their annual report of 2022 cases. So like I said, we're wrapping up our last filings for those cases, and really the ones that are left are the mass stranding events. We've been trying to get some more information on those from the domoic acid bloom that happened in Southern California, where we had over 1,000 pinnipeds. So that was mostly California sea lions, a few other types for seals. And then we had over 100 dolphins. So unfortunately, that was a mass stranding that was really upsetting to see, but we want to make sure we capture that and get it in the tracking databases. And then for some presentations, we've had an opportunity to share our work and also the grander efforts of California. As was mentioned by Yana, thank you so much. In US EPA Cyano Symposium, the whole work group presented in the state and tribal feature. So that was a 4-day workshop, and each day a different state or tribe was featured. On another day, we also had the wonderful work presented by Sarah Ryan, one of the co-chairs here, with the Big Valley Band of Pomo Indians and the Environmental Protection Department efforts. So that was really highlighted nicely. And then the California HAB illness work group was on another day. That was October. In November, I got to present for the California Poison Control System folks. So making sure that their coding is in line with getting those potentially HAB related cases to us to interview. And then just recently, yesterday, California had their first One Health Symposium. So as we have the webinar series from the feds, as our partners just mentioned, that are forthcoming this year, the first one being today, please keep an eye out for California Department of Public Health's feature, where we're going to have One Health interests across the board. So HABs were featured yesterday. That was also recorded, and our colleague at CDPH, Thomas Hayashi, was the lead presenter for that. So that is the update. Thank you so much.
Thanks for representing us, Shannon. Awesome job. And now we will move on to the mitigation subcommittee update. Hugh, are you available?
Yes, can you hear me?
Sure can.
You want to throw my slide up there for me, please?
Oh, sure. Give me a second here. Did you send that to me via email? Do you have it available?
I don't, but I can read it off if we don't have it available.
Let's see.
It was in the email I sent yesterday.
I don't see it in that email. Would you just read it off for now? Sorry
Sure,
about
no
that.
problem. First off, good morning, everyone, and thank you for the outstanding updates I've heard this morning. Really some great work going on. I represent the mitigation subcommittee, and my partners in co-chairing are Bill Taylor, Dave Karen, and I also have Jake Iverson from the State Water Board, replacing Katie Fong this last year. Where the four of us make things happen on a monthly basis. We meet every third Thursday from 2:30 to 4, the months March through October, and focus mainly on, the HABs map, current things going on, and mitigation successes, stories, and emerging technologies. So today's report, I'm talking about our goals set forward for 2024. And we have a few water bodies that we plan to study and evaluate in 2024, which are Lake Temescal and Lake Anza. The agency East Bay Parks, our lead investigator on that will be Bill Taylor. Lake Wildwood up in the Sierras outside of Sacramento, a private HOA group, and that lead will be Dave Karen. And myself, Hugh Dalton, I chose Pinto Lake down in Watsonville. So the team chose these waters to evaluate because of their relative close proximity to our homes, the abundance of historical water quality data, and the knowledge that each agency is working with a California certified lake management consultant. We plan to provide unbiased and independent evaluations as a three-person team. We plan to document their mitigation strategies implemented over the past 10 years, their near future plans for HAB mitigation and water quality monitoring, along with a comparison with other mitigation solutions. Agency decisions, as we know, often have a long lead time, often requiring 1 to 3 years or more. An example of that at Pinto Lake was getting a grant in 2014, doing an alum treatment in 2017, and then also having a requirement to wait so many years before actually doing a different mitigation, which I think was right around 5 years of time. In addition to Lake Temescal and Lake Anza and East Bay Parks, they also manage other lakes and over 600 ponds. Lake Wildwood staff and homeowners are motivated to improve their water quality in order to proactively minimize any future HAB events. And Pinto Lake, of course, is interested in its ongoing mitigation solutions that they're ready to take on a new and different mitigation in the coming years. A little background on Pinto: it's an 8,000-year-old lake. It's about 15 minutes down the road from me. They had the most serious toxin event in California in 2007, where the lake measured nearly 3 million parts per billion of microcystin that flowed into the Monterey Bay and killed some sea otters. So likely our group here is in response to something as horrible as that, and documented as probably one of two of the highest toxin detections in the world. So with that said, the focus for us as a group isn't to solve these problems this year, but in order to study and evaluate how mitigation solutions are drawn out, the permitting process, and the success or lack of success thereof. Open for questions. And again, we meet monthly, third Thursday of each month. And thanks to my co-chairs if they have anything to add today. I would want to add one more thing about bioremediation techniques. It's an emerging technology for mitigation. Right now, currently, people with ponds are able to buy pelletized bacteria and enzymes to treat small water bodies, say like 1,000 square feet at a time. But for larger lakes, there are now innocuous bacillus bacteria generation modules that people are using for municipal and industrial wastewater, and also being applied to fresh water. I know in California it hasn't been used yet, but we're hopeful that we can find a pilot study for that in the coming years. And that's all I've got for today.
Thanks, Hugh. It looks like you've got a question of how they can get in touch with you in order to join your meetings.
I'll just throw my email down and they can get a hold of me. Otherwise, it's our state water board contact is Jake Iverson with the water boards, and sometimes things go through him, but I'd be glad to give my email out.
Perfect. Thank you. Okay, and now we've got our final subcommittee update from the Benthic HABS Guidance Committee. I think, Carly, were you going to give that update?
Yep, that's gonna be me. Hold on, let me I'm getting the screen up for you. It's just a couple slides, not anything crazy, but so the Benthic HAB subcommittee reconvened for its first time in a while in May of 2023. We have a very strong, small but mighty team, actually. So the committee team leads are including the rock stars from Region 1 who've been studying benthics for a while, Rich and Mike, and then Marissa and I at State Board. And we are getting a lot of support from Jamie Smith from SCORP, who you guys probably know because she's a co-chair for this as well, as we have a contract deliverable for her to kind of start looking at benthic SOP development and writing a technical report for us to hopefully lean on as we move forward with all things California. And I know as well, Dana primed it pretty awesome for us in terms of the US EPA ROAR study that Region 1, Marissa and I, and then also Region 5 got to participate in. And they also have some of their team that's working on that study participating in our work group, as well as folks from Utah and Nevada. So it's pretty awesome so far. We've had 3 meetings starting this fall to really start diving into things. And we're having about monthly meetings through May, at least from what we've kind of forecasted at this point. We've been making some pretty decent progress, but we've had some really amazing lengthy discussions too, which have made us pause and think a little bit more diligently about what we're doing. So with that, if you're interested, you can just email the CCHAB reports, water boards email, and we'll definitely add you to the list. So just to talk about what we have been working on, is we've been really working on expanding our current benthic guidance at the statewide level. As you may know, it's presence absence in terms of our advisory status, and then also our collection methods. I know Bill made a comment about if we're designing benthic sampling instructions specific for lake HABs, and I think our group is going to go, or at least we're considering and potentially pursuing the process of whatever we develop, we're going to at least put caveats at what would be applicable for sampling for lake HABs, because we don't want to have it be completely absent from our guidance. So we are going to try to incorporate the best we can based on our limited scope. So hopefully we'll have something that'll be at least helpful for those folks dealing with some of those lake-wide benthic HABs. So the goals of this benthic guidance is to determine a little bit better how we want to respond to them, how we want to sample for them, and then how we want to also put up signage or change any signage for them. So currently we've been looking at the latest science and what other folks are doing, including the US EPA ROAR study, and existing protocols, and then looking at the different indicators and which ones that we kind of want to pull in and envelop into our guidance document of whatever we decide to pursue. And we're still really in the meats and potatoes of really kind of dialing in what we want to focus on and where we want like the scope to not creep. So we don't have anything exciting to present to you as of yet, but I'm certain by the spring we'll have a lot more to bring to you for discussion as we really start diving in. So yeah, if you're interested, you can reach out to me or the CCHAB inbox, and we'd love to chat with you about it. And that's all for me.
Thank you so much. All right, we have arrived at our lunch time. We will re-adjourn at 12:40 for 2 more really exciting talks, so please be sure to come back for that. And until then, enjoy your lunch. Thanks, everyone. Okay, everyone. So our next presenter is Dr. Keith Lopton and Zach Lafferty. And thank you so much for coming and presenting to us. They're going to be talking about a review of algal toxin exposures on actually reserved federal lands. So that's going to be really, really cool to see what they have to say. If you wouldn't mind, go on ahead.
All right, thank you very much for inviting us to be here. Zach and I are going to tag team this talk today, and appreciate the opportunity. So this effort culminated in a journal article with the citation down at the bottom of the screen here. The DOI number is listed if you would like to read it. It's a little bit of a lengthy read, I'll admit. This effort took us about 3 years or so to get from start to finish, give or take. And we'd also like to thank our contributors that worked with us on this, several from USGS, and then Fish and Wildlife Service, National Park Service, and the U.S. Army Corps of Engineers. And the funding that supported this effort was supplied by the USGS Ecosystem Missionary Environmental Health Program for our USGS colleagues. So I believe even still up to this time, there's not been an additional accounting of toxic algal bloom effects on federal lands. And so this was a bit of an effort to try to navigate for multiple reasons. One of which is just the sheer number of lands that exist and the amount and how much they change hands on a daily basis. I was surprised when we learned that. But approximately 259 million hectares is in public, tribal, or Native American or military usage. 75% of all federal land is managed by the Department of Interior bureaus, which USGS resides in also, but we don't have any land management responsibility. But the bureaus within Department of Interior include Bureau of Indian Affairs, Bureau of Land Management, Bureau of Reclamation, National Park Service, and U.S. Fish and Wildlife Service, with Bureau of Land Management holding a majority of the lands. As you can see in the map, in the lower right-hand corner, a vast majority of our large tracts of lands are in the western half of the U.S., but we have lands, federal lands, as we all know, spread across the country. So the purpose of federal lands is varied. Some people don't realize that it's not all preservation of national treasures and sensitive ecologic areas. But it's also used for accessing natural resources for extraction of energy, minerals, wood, grazing, drinking, and irrigation water, flood control, recreation, subsistence hunting, and abatement of invasive species. And so a lot of these are handled through legislative actions. In fact, all of them are that direct how these lands are to be utilized and held in public trust for public use, and what those limitations may be for the different lands. For our study, evaluating algal toxin events on federal lands, we focused only on the harm due to toxin production only. So we were not paying attention to aesthetics like taste and odor, or it looked bad, those types of things, and we weren't paying attention to socioeconomics, so just toxin. The data that we used was obtained mostly between the 1990s to present, but some was from before. We had to really think about how we were going to go after information, because as a federal agency, we're subject to the information collection rule, which means we can only ask 9 states the same question without going through a 2 to 3 year process, culminating in a Federal Register notice. And that's legislation that we have to follow. So we couldn't go and call all of you and say, hey, can you give us all of your databases for this effort, without that process. And so it would have doubled our time. So what we elected to do is to go through peer-reviewed literature, government reports, publicly available state and agency databases that we could find, communication with federal agencies and bureaus, and then accessing databases of the USGS National Wildlife Health Center, where that particular center receives specimens from other federal agencies like Fish and Wildlife frequently when it's suspected of, an animal mortality is suspected of a particular causative agent that they're researching. So as part of this effort, as you can imagine, and some of you have already alluded to or spoke of today, just defining what an event is can be pretty difficult. And you heard Megan Coffer talk about, how we, would go about deciding temporal frequency and spatial extent and all that. And so just from the abiotic aspects, it's difficult. When you start throwing the biology in, it really can make things even more complicated. And we'll talk later on about some of those complications, areas that we might be able to approve. But we decided to divide things up into 3 tiers. The first tier is that of occurrence for algal blooms and or toxins below a threshold of concern. And that threshold of concern was usually a state or local threshold. Most federal agencies default to the states and work closely with the states on those issues. And also, if no threshold existed, it would have fallen in the tier 1 category. Tier 2 would have been the second tier, encompassing the events in which a local or state threshold was actually exceeded, triggering an advisory, warning, or closure. Tier 3 is where we introduce animal morbidity or mortality that's believed to be caused by algal toxin exposure with various levels of clinical and toxicological evidence. So you may not remember, but the timing of the case definitions that were produced by CDC was also towards the end of this effort. And this effort actually helped inform some of the feedback we gave CDC on those case definitions. So we did not use their case definitions, but I think they're somewhat similar. And then it's challenging to identify toxin effects in animals. And so this includes remote areas that make routine surveillance difficult, lack of resources to gather animals and determine causes of death, a lack of field-based case definitions to readily identify potential toxin poisoning cases. We have to deal with predation of sick animals by other wildlife. We have a range of animals that are geared towards that predation activity: coyotes, wolves, etc., scavengers, even birds, they're scavengers. Many of the animals are migratory in nature, and they move between public and federal lands with frequency. All of this culminates in that we expect that what we're reporting is actually an underreporting of toxin exposure in the United States on federal lands. And I'm going to turn it over to Zach Lowry now for several slides. Zach, are you with us?
I am. Can you hear me?
Yes.
Okay, good. So what we found was that at least 11% of federal lands have algal blooms and/or toxins present, dating from the 1990s to the present. Well, at least when this article was written. 11% is a weird number, and I want to tell you how we got there. And it's weird because it's, how do you define that? So what we did was we looked at an area of federal land controlled somewhat locally by an agency. For example, Tuskegee National Forest in Alabama is a national forest, is one unit of the 174 national forest units. Voyager National Park is a unit of the National Park Service. It is one of 88 of those units that are managed by the National Park Service. So if we look at this chart, we can see on the y-axis, we have percentage of effective land units. Oh, I should also say, let me step back for just 1 second. Voyager National Park has several bodies of water present on it. If there was toxin or algae present in any of those water bodies, that counted towards Voyager National Park at that unit. So if you look, so the y-axis is the percentage of effective land units. and the x-axis is different federal agencies or bureaus. The numbers above the columns are the numbers of affected units, and the denominator is the total number of units in that bureau. So, for example, the Bureau of Reclamation has 29 of their 338 units were affected by algotoxin. And then if we total them up, we get 216 affected units out of 1,944. Now, I would like to point out an additional thing. The National Park Service is unique when you look at federal agencies that have land management responsibilities. The National Park Service not only has large tracts of land like Yosemite and other beautiful national parks, but they are also, yes, they are, as much as to answer the question that just popped up on my screen. Yes, they are. We treated tribal lands as part of Bureau of Indian Affairs, just for a lack of an ability to differentiate them in other ways. And there's complications with that as well. So to get back to national park, the National Park Service not only has these large tracts of land, but also they do historically important or culturally important sites, such as here in Kansas, we've got the Brown versus Board of Education Courthouse, which is a national park site. Stonewall Inn in New York City is a national park site. However, only 88 of the sites that the National Park Service manages have water bodies, extensive surface water that would be susceptible to algal bloom. So we used 88 for this study. Another important thing is interjurisdictional overlap. So there were cases in which, say, there is Army Corps of Engineer reservoir was created. On the banks of that reservoir were some tribal land in one part of it. In another part of it, the Fish and Wildlife had a refuge set up. So there's an algal bloom with toxins that caused some sort of advisory to be put in. How do we account for this? So what we chose to do, realizing that no answer is perfect, what we chose to do was whoever controlled the water, in this case, Army Corps of Engineers, it would be put into their bin, for lack of a better word. We would consider that their unit, even though both in this case, the tribal land and the Fish and Wildlife Refuge may be affected by this bloom. But we didn't want to overcount things, even though there may be some validity to that. Keith, would you go to the next slide, please? So if we look at algal toxin events categorized by tier, by federal agency, and we can look at this graph and look at percentage of events in each tier. Just to remind you, in blue is tier 1 events, which is the events of occurrence. The orange columns represent tier 2, which is exceedance tier. And tier 3, which is the gray bars, are events in which there were animal mortality or morbidity. And you can, and the purple numbers above the columns are how many events occurred within that bureau or agency. So for example, in BIA, there were 18 that we found events, and then the percentage of the events, did they fit into tier 1, tier 2, or tier 3, are shown beneath that. One thing that jumps out is tier 2 is looks like the dominant tier in most bureaus, and this makes sense because if someone goes out and takes a measurement and it's below a level of concern, the concentration of a toxin or the number of cells or however they're going to judge that particular water body, that doesn't necessarily make it into the literature or a report. It may just be filed. But when there is an exceedance, which is tier 2, that does tend to stay around a little bit more. It gets it gets put into databases in the literature, etc. One interesting thing to think about here, or to look at, is the number of events in each federal bureau or agency. The National Park Service and Fish and Wildlife Service seem to have a lot more events compared to other agencies. I do want to point out that both the National Park Service and Fish and Wildlife Service have ongoing event-based monitoring system programs, which not all the other federal agencies do. The National Park Service and Fish and Wildlife, they have parks that are all freshwater, and they have parks that are all marine, but they also have some that are mixed. And so having that kind of variety increases their possibility of having exposure to algal toxins and etc. Also, National Park Services and Fish and Wildlife refuges are very well visited, and what you might have is that you have people going out, seeing these beautiful places, and then seeing an algal bloom, which gets reported to a ranger or someone else who works there, and then then that triggers an investigation, which is then reported later, which may not be the case for all the other agencies. So Keith, would you go to the next slide? So we thought it would be informative to look at this information at the state level, keeping in mind that, as Keith talked about earlier, the federal lands are not equally distributed amongst the states. And so there may be more federal lands west of the Mississippi, certainly, than east of the Mississippi. But let's walk through this map. So the deepness of color of each individual state is based on the total events in that particular state. If it's very light gray, there may be 0 to low events, and as it gets darker, there are more events present. The tiers are indicated by different icons. If it's a blue circle, that indicates tier 1. Tier 2 is indicated by a yellow square, and tier 3 is a red triangle. The size of the different icons indicate the number of events that were in that tier in that state. They are not all equal. The gray range goes from 0 to 620 events. Tier 1 is from 0 to 95. So the smallest, well, if it's not there, it's 0. So the smallest one starts at 1, the largest one ends at 95. Tier 2 goes from 1 to around 600, so the smallest, and then tier 3 is from 1 to 20, yeah. So if you look at this map, one of the things that jumps out is that a lot of coastal states have the largest number of events, and that is true. However, a lot of those coastal states are, of course, exposed to marine toxins versus, in addition to cyanotoxins in freshwater. You're looking at California, Texas, Florida, and Maine, and New Hampshire and Oregon. But also the central states, there's some variability in that, as Keith mentioned earlier. There's no universal advisory level for all states. So each state sets its own advisory level. Some set it, or may not have any at all, right? So the smallest, one state may have a very low concentration, which triggers an event, an event posting, versus another state which has a higher concentration required to trigger a posting or an advisory. And also some states have public-facing databases that are able to be searched. And so if I came across those databases, I used them. Additionally, there are some researchers who are located in certain areas that study a specific coastline or water bodies within a state, either governmental agency or an academic researcher. So that can show up in the literature as well. All right, Keith, will you give me the next one, please? Okay, good. So we're going to pivot a little bit. We're going to talk about trust species. Trust species are certain species of animals that are composed of migratory birds, threatened species, endangered species, marine mammals, other species of concern. They're protected due to legislation or treaties that the United States has in place. These are protected whether they are on federal lands or not. So in this case, in the previous case, we were discussing like tier 3, for example. We were discussing events that occurred on federal lands. When I'm discussing trust species, it happens either on or off federal land. Anywhere that a trust species was exposed to algal toxins, we tried to account for it. You can get rid of trust species. I'm responding to the text as I see them. So if I don't answer your question, I'll try and get back to you in the text later. If you go to my paper, they are in the supplemental information. I tried to list all the trust species. I did list all the trust species we found and either the record of them, either in the literature or government record. So what we found was that and different federal agencies have authority to manage and monitor trust species to ensure that the natural resources and natural habitats are available for their benefit. These federal agencies include the Fish and Wildlife Service, the National Park Service, and the National Oceanographic and Atmospheric Administration. So what we found in the literature was that 68 different trust species have been exposed to at least one of 6 classes of algal toxins. If we look at this map, we can see, once again, the color of the state, the deepness of color is how many trust species were affected in that state. The white areas or transparent areas are 0, and anything with any gradation of gray going to deeper color has trust species present. The icons that are there, they're triangles. The smaller triangles to larger triangles is the number of species affected by toxin, and depending on the toxin is the color. So brevetoxin is this green color, saxitoxin is kind of a salmon color. Golly, I can't see it. My eyesight's bad. But anyway, we got, let's see, microcystin kind of purple, oxalic acid is green, domoic acid is yellow, and the toxin that causes avian vacuolar myelinopathy is in blue. So you can see if we look at this map. Oh, thanks. Most of the states in which we have trust species being exposed to algal toxin are once again on the coast. California, Florida, along the east and west coast, Texas, of course. And the toxins are different depending on the coast a little bit, but domoic acid seems to be present everywhere. Brevetoxin, of course, is going to be more prevalent in the Florida and a little bit in Texas. But the middle states or Midwestern states are not free from this. We see microcystin present in Iowa, Minnesota, and North Dakota, affecting trust species. And of course, Alaska is present here as well with domoic acid and saxitoxin. Keith, can you move to the next slide, please? Thank you. So to further kind of break down this and discuss some of the issues, or not issues, complicating factors. So this is the toxin that's present and the different types of general trust species that we're looking at. And we can see that marine mammals, of course, constitute a lot of the exposure of trust species to algal toxins. Almost every class, except for Keith, can you say that last toxin? I have trouble with that. That's the avian toxin. But I'll try and say it. Why would you go?
A toxanotoxin.
Icactanotoxin. Thank you. So, which affects both raptors and different migratory birds. Migratory birds is also very present, as well as threatened and endangered species and species of concern. We do see in the literature that either concurrently or sequentially, that we are seeing trust species being exposed to different algal toxins. So there's examples of bottlenose dolphins, which you can see are exposed to 4 to 6 of these 4 of the 6 toxin classes, and also the manatees. These are creatures that can go kind of from the marine environment into more of a coastal environment where you may see cyanotoxin runoff from freshwater interior lakes, as well as other toxins. We do see, in addition to marine mammals, migratory birds, there are reptiles that are affected. So these are going to be under threatened and endangered species, and fish, of course. So Keith, I think it's back to you.
Thank you, Zach. So in summary, 11% of federal sites overall were observed with some level or tier of potential harmful algal bloom or toxin exposure of some type. All agencies discussed have some reported issues. Nobody's immune, from this phenomenon, but that's not a shock at this point. Earlier on, 20 years ago, some people weren't even convinced this was a national phenomenon, let alone a global phenomenon. I think that ship sailed some time ago. Effects on trust species, at least 68 trust species were affected by algal toxins that we know of. Effects are throughout US in at least 18 states. I'm sure there's more than we've accounted for. And I'm sure there's many of your colleagues that have information more than what we have today. Exposure to multiple toxins sequentially or concurrently. One thing we haven't mentioned that could also convolute mortality and illness cases is everything else that the animals are exposed to. Rodenticides, lead, any number of pesticides, herbicides, insecticides, etc. So it really is a mixture, and that gets us into the One Health framework. And then animal movement and migration connects private and public land exposure. It's possible that we could find an animal in a pristine water body on public or private land, but this exposure occurred elsewhere. And so that would convolute the accounting for this phenomenon as well. So some of the needed improvements that would be beneficial if we wanted to actually use animals, different species exposed to algal toxins, are they good sentinels for ecological and human health, harm due to toxin poisoning? And part of that is we're always looking for early warning. And with the public facing aspect of many of these agencies and their lands and how utilized they are, national parks has gone to a reservation system that they're getting so many visits. We really are looking for any angle to, provide that early warning for folks. And so it really needs to be a multi-pronged effort. Needed improvements, harmonization of taxonomic nomenclature of algal bloom organisms. That's a tough one. It's been going on for a number of years. Standardization of state and federal action levels would make categorizing cases easier. I don't say that as a criticism, but there may be some very logical reasons for having different thresholds. It just represented a difficulty in showing an even playing field for this study. And we didn't always have access to the data. It may just tell us that it was an advisory or a warning or a closure, depending upon what information we were looking at. Better case definitions, no offense to the efforts that CDC and our colleagues and ourselves worked on. There's always room for improvement, especially on chronic exposure. It's so difficult. So many other things are coming into that exposure space that I mentioned earlier. Standardized response to animal deaths. We don't have the resources to respond to everything or find everything. But there's not enough boots on the ground, let alone people trained to look at that. Zach's point about citizen science and how warnings may come to agencies through public visits is an important one to keep in mind, and it may be an opportunity for education. Systematic and routine monitoring of algal blooms and their toxins. One of the most difficult things that we deal with in trying to define cases and events is by the time we know something happened, the water quality has already changed. And so if we had more of a systematic or routine monitoring, we might have a better chance at capturing what the water quality circumstances were at that time of the exposure, should the animal be found in time. And that's an issue as well. There's only a certain window for when the necropsies can be performed on the animals before their carcasses are no longer viable. Better diagnosis for understanding chemical and pathogen mixtures effects related to algal toxin susceptibility, especially for chronic toxicity I mentioned earlier. But we don't currently have real-time toxin detection capability that our veterinary colleagues can utilize during necropsies to say, oh yes, this tissue was indeed exposed to cyanotoxins, it was a saxitoxin. And so they would have data-directed medical information to correspond with the symptomology. And that ties right into the biomarkers of exposure as well. Do we have things that we can hang our hat on where we absolutely definitively know it was cyanotoxin poisoning versus something else? Some of the symptoms are pretty generic, like in the case of saxitoxins, it may just be respiratory suppression. And there could be numerous etiologic agents that cause that problem. And then assessing economic impacts. If we understand, what the role of animals are, what their value are, and ourselves included in that scenario, and what these lands are valued at, for our usage and varied usage, I think it starts putting the problem in context. And so I think that's an area we could improve on as well. So with that, we'd like to conclude our talk. And again, the journal article citations at the bottom, and happy to field any other questions that there might be, if there's time.
Yes, there is time. Thank you so much for your presentation. Does anyone have questions? Feel free to unmute and ask now. Seems like you guys answered a couple that were in the chat already. Oh, it looks like Christine dropped one in the chat here.
That's what I'm reading right now.
Oh, okay, great.
Yeah, how will this information be used by the federal agencies? Will they be receiving or asking for funding to do monitoring among their lands they manage? Perhaps the agencies can use the satellite imagery from Cyan project to help prioritize areas within their purview. For example, we've noticed that some federal agencies like USAID here in the West do not do any kind of routine monitoring. How will this information help them address these issues? Boy, there's a lot to unpack there, but a lot of good information. So thank you for that, Christine. In terms of how the federal agencies will use this information, I think it puts a low bar on how bad the problem may be in terms of our context of underreporting. And I think it starts pointing out places where we might be able to make improvements in the future. And so I think I'm very comfortable at that level. In terms of, will they be asking for funding to do more monitoring among their lands they manage? Funding has been increasing steadily over the past several years to do more and more work on a variety of different areas for many of the agencies, including agencies that don't have land management responsibilities. And part of that is through efforts of groups like yourselves, IWG 4-H or HBARCA, and various other activities where our legislators are made aware of different problems. And so those are some of the things that I think I see happening on the landscape currently. In terms of using satellite imagery from the Cyan project, we're actually working on a new effort that I just spoke with our team about a couple of weeks ago. Actually, we're looking at migratory birds and what it might look like if they were pond hopping or lake hopping through a migratory pathway using the Cyan imagery. Obviously, we're not going to get the small water bodies, and so we're excited for Cyan 2.0. But we are trying to take some steps along that effort. It won't be a complete picture, but it'll be a step in that direction. And in terms of agencies that do or don't do routine monitoring, I think we could probably say none of us on the federal level are doing routine monitoring everywhere just because of sheer amount of resources it would take. And routine means different things for different problems, right? And so it depends on what the question is. And to be able to observe animal effects, that would be a very resource intensive routine monitoring effort, to say the least. Sarah Ryan, that was my question too, also shared with state agencies and tribes. What do you mean by the last part, Sarah?
How this type of evaluation that you're doing is being shared with states and tribes.
So it's already been published, and then presenting at groups like this are one way that we get that information out. If you have other suggestions, we're happy to entertain those ideas as well.
Well, I guess it's not just shared, but also are there expectations by the federal agencies about how this information is going to be used within like Clean Water Act programs throughout the U.S.?
I can't speak to that being in a non-regulatory agency. I think that would fall more into EPA and NOAA jurisdiction, especially on trust species or U.S. Fish and Wildlife. So if somebody's on the call that would like to address that question, feel free.
Okay, thanks so much. Any other, we could take one more question if we have another one. All right. We'll definitely be following your research in the future because, sorry, did someone else just unmute themselves? Maybe that's feedback. Okay.
Thank you so much.
Yeah.
Thank you.
Okay. Thank you. And our final presenters today, we have Dr. Ellen Priest and Janice Dr. Janice Cook as well. And they are going to be presenting today on microcystin in Delta shellfish. I can see your slides. They look good, Ellen.
All right. All right. Well, thanks for having me. I'm going to present some research that we did in collaboration with Tim Otten at Ben Genetics, and Janice and I are worked on this together from Central Valley Regional Water Quality Control Board, and then Rafe Kudela at University of California, Santa Cruz. This is a Proposition 1 grant funded project, and we're just wrapping up all of our findings and analyzing our data. And so I'm presenting that information to you today. And I'm going to present really on a 2-year time frame from July 2020 to about September 2022. So just a quick overview about microcystin in shellfish, since that's the main topic of my talk today. So the most common CCHAB-forming cyanobacteria genus is Microcystis, and this cyanobacteria is often producing microcystins. There's growing evidence that these microcystins can accumulate in a variety of shellfish. And microcystins are a particularly stable toxin, and so that's one of the reasons that they're able to persist in the water column and then also accumulate in shellfish species. They've been found in a variety of different shellfish, ranging from clams, oysters, mussels, crabs. Filter feeding bivalves are particularly good at accumulating microcystin concentrations above ambient levels because they're filtering all of the water that's in their zone of influence. And so if these organisms are accumulating toxin, they represent a potential hazard to people that are consuming the shellfish as part of their diet, or to fish or other species that are feeding on the organisms. So the focus of my talk is the Sacramento-San Joaquin Delta, which is part of the larger San Francisco estuary. And so I wanted to present a couple of past studies that have looked at microcystin in shellfish in the San Francisco estuary. These studies have all focused on the saltier portion of the estuary, and our study is focused on the freshwater portion, which is also referred to as the Sacramento-San Joaquin Delta, or Delta. But just some evidence for why we had interest in looking at microcystins in the freshwater portion. On the left is a figure that looks at microcystin concentrations in Potama corbula, and these are an invasive clam that are found in the saltier portion of the estuary. And you can see on this diagram, this time series, that in Sassoon Bay, there were relatively high concentrations of microcystin detected over the time series. Another study, and these next two studies were both out of the Kudela laboratory. Wild marine mussels tested positive for microcystins during every month of the year in the western portion of the San Francisco estuary. And then Peacock and others placed caged mussels across the estuary, and a couple of the cages were just within the legal boundary of the Delta. And they found that the mussels, 82 to 100% of all of their mussels accumulated microcystin. So there's really good evidence that there's kind of this persistent microcystin contamination of shellfish in the San Francisco estuary. So in the Delta, so that's the brown boundary on the right side of this map, the cyanobacteria harmful algal blooms have been an annual occurrence since 1999. At the same time, Asian clams, so these are corbicula, and basically the Potacorbula, they are all over the saltier portions of the estuary, and the corbicula are found from the transition to the freshwater portion of the estuary. And they are everywhere in the Delta, and they can be in really high densities. There's papers out there that have shown that they have a lot of ecological impacts on the Delta. One of these is that they've replaced other common prey items for native species. And both humans and fish species are known to consume these Asian clams. Another introduced species is the signal crayfish. These are a benthic dwelling
omnivore that will eat just about anything, including Asian clams and mussels. And crayfish are harvested to sell in restaurants, and organisms, other biota are also consuming these crayfish. So our project goal was to determine if microcystin, and we also looked at a subset of samples for saxitoxin, are a stressor on food web and fish, including managed species. So this Proposition 1 grant was specifically interested on the impacts of toxins on fish and managed species. And so that's one of the reasons that that's part of our project goal. And managed species in this case is really thinking about white and green sturgeon. And then a secondary project goal was to determine if microcystin and saxitoxin and shellfish presented a risk to human consumers. So I'll show a map in the next slide of the different sites that we chose, but want everyone to know that they are sites that are known to have harmful algal bloom issues, but they aren't necessarily the hotspot areas in the Delta, because we coordinated with the DWR and CDFW sturgeon team to make sure that our sites were also representative of known sturgeon habitat. Technically, juvenile sturgeon can really be anywhere in the Delta, but there are certainly certain areas where they're more known to feed. And so we were trying to kind of meet a balance between where there could be these harmful algal blooms and then sturgeon hanging out at the same time. And so these are the 10 sampling sites that we came up with. Green sturgeon in particular use the Sacramento River as a migration pathway, and Sherman Lake is a big feeding area for them. But we predicted that the Cache Slough and Sacramento River sites would have less mussel clam contamination than the central and southern Delta sites. For our field methodology, we sampled for 2 years, starting in July 2020, and that was twice a month from June through October, and then once a month for the other months. We used a Ponar sampler, which you can see the picture here on the bottom right. And we pulled up sediment samples, and then we sifted them through a crate, which is kind of next to the motor in the boat picture, until we had about 50 to 100 clams per site. So it depended on the site and how well the Ponar was working for us on the particular day. But it usually was about 3 different hauls of sediment to get enough clams. For crayfish, we set out crayfish traps, and those traps have to be baited and they would be set out overnight. And we were not nearly as successful at collecting the crayfish as we were the clams. We often would have empty traps. Several traps were stolen throughout the process, but we did collect some, and I'll be able to present data on that. And then on all of the sampling dates, we also took grab samples so that we could look at toxins and other parameters in the water. For the lab methods, the picture here shows one of our sites on one date. So that's all of the clams that went into a single composite sample. Tim's lab, Ben Genetics, they removed the shells, they composited the samples and freeze dried them. And then they followed established procedures and extracted the freeze-dried tissue with methanol. They measured microcystin on every sampling date, and then they measured saxitoxin on 3 of those sampling dates. In addition, and they used enzyme-linked immunosorbent assays, or ELISAs, to colorimetrically detect the toxin. We set a subset of the samples to UC Santa Cruz for LCMS analysis, where we looked at 8 microcystin variants and then the MMPB chemical. And the next set of slides is me discussing the comparison between these 2 laboratory methods and some things that we learned and were slightly surprised about as we're analyzing the samples. So method comparison between the ELISA and the LCMS. So just a quick overview for those of you that may not be familiar with these different analytical techniques. So the ELISAs are commonly used. I've heard them mentioned several times in today's talk, and they're really good for measuring microcystins in water, and they've also used
are commonly used for more complex matrices such as shellfish and fish and sediments. There are a variety of commercially available ELISA kits. Some people also can make their own. And the one that we used in our study was a polyclonal ADA ELISA kit. And the ADA is a unique side chain. It's on, so there's over 250 microcystin variants, and that ADA is present on all of them. And so this ELISA kit allows us to look at all of the potential microcystin variants that may be present in a sample. And the ELISA kits are really great for rapid and relatively inexpensive analysis. There has been evidence in the past that they can be prone to matrix effects for shellfish and other fish and animal products. And then the other method that we use is the high performance liquid chromatography coupled with mass spectrometry. And so this can be used as a tool to identify and quantify the specific microcystin congeners. It's highly specific and less prone to matrix effects. However, it can also experience matrix effects, and I will get into that in our next slide. The standards for this are limited. So I mentioned there's over 250 microcystin variants, and there's only standards for a few handfuls of these. And so it may underestimate the total amount of microcystin in a sample. So the other thing that we looked at was we also used the LCMS to analyze the MMPB. This is a chemical that is an oxidation product of microcystin. And so it allows a quantitative analysis of total microcystins. Unlike the ELISA and the LCMS, the routine, it is able to look at both the freely available microcystins and then the microcystins that are bound to proteins. It's expensive and it's more time consuming to do the processing. It also remains unclear if those bound microcystins are a risk to human consumers or if it's just the freely available microcystins. All of our samples were first analyzed by ELISA, and then we subset a range of different toxin concentrations to Santa Cruz for the LCMS analysis. We sent 32 of our 339 clam samples and 4 of our 30 crayfish samples. All 36 samples were analyzed for the 8 microcystin variants that they were testing for. And then we did only 10 samples with the MMPB method, 9 clams and 1 crayfish. And our first set of results, they didn't match with the ELISA results. Some of the LCMS results were much higher than the ELISA results. Some of the LCMS results were much lower than the ELISA results. And so we had a number of discussions with UC Santa Cruz to try to figure out why there was this discrepancy between our 2 methods. They've been using this methanol extraction method from Macabry and others, 2009, which is actually, I believe, a Department of Fish and Game paper. And as one of the cleanup steps, they use these Baker Bond C18 solid phase extraction columns. I have pictures of what those look like on the right. And basically you have your extract and you want to filter it through that so it's clean enough to run through the LCMS. And talking with Rafe at UC Santa Cruz, he had done an Asian clam study for a different part of the country and was starting to suspect that these Baker Bond C18 columns were not doing a good job cleaning up the Asian clam samples, even though he routinely uses them for a number of other animal tissues. And so he had read in New Zealand, they had switched to using this Strata X solid phase extraction column. They look the same, but the materials within them are a little bit different. And the Strata X column has a finer particle size. So the sample flows through it more rapidly and more quickly. So maybe there's less potential for things to get caught in the column.
Once he cleaned up all of the samples with this different column, we found really good agreement between the ELISA and the LCMS. And so this was a pretty interesting finding that, especially in the case of crayfish and Asian clams, that the standard column really wasn't working very well for LCMS analysis of our samples. This is a figure showing the ELISA samples on the x-axis and then the LCMS samples on the y-axis. I think there's pretty good agreement. There's obviously a couple outliers that are skewing the R-squared, but overall we're pretty happy with the agreement between the two methodologies. And then I wanted to show you the table of the three samples, or the three analytical approaches that we use with the 10 samples. So we have ELISA on the far left column, then LCMS with the 8 microcystin variants in the middle column, and then the MMPB method on the far right column. And there's not a perfect agreement here, but in general, we found that the LCMS with the individual variants detected less microcystins than the ELISA, possibly because it was missing some of the congeners. When we ran the MMPB method, it had pretty good agreement with the ELISA, but in some cases it had higher toxin concentrations, and this was likely related to it measuring both the bound and unbound microcystins. Some other notable findings is that only 4 of the 8 microcystin congeners were detected. The microcystins YR and LR were present in most samples. Over half the samples had the microcystin RR, and only 5 samples had microcystin LA. Okay, so now I'm going to go into some of the results about the microcystin in shellfish and water, and this will be just the ELISA results that I'm presenting in the next few slides. So first, this is looking at on all of the clams in a single box plot, all the crayfish, and then all the water samples. So we had 339 clam samples and 339 water samples within this box plot, and then 30 crayfish samples. And then we have 2 health-based advisory thresholds that we're looking at. The solid gray line is the California action level. Note that it has been adjusted to account for our samples, which were in dry weight. The value given in the literature is based on organisms' wet weight, but all of our organism findings are reported in dry weight. And then the dotted line is the World Health Organization tolerable daily intake. And what you can see is that for our clam samples, we only had 7 samples that exceeded the California action threshold, and only 4 of those exceeded the World Health Organization tolerable daily intake. For the crayfish, we had 2 samples that exceeded both of those thresholds. And then our concentrations were quite low. We never exceeded the California warning threshold of 6 micrograms per liter at any of our samples. So on this figure, these are the 10 sampling sites. And on the left y-axis, it's the microcystin in clams, and that's the gray shaded area. And then on the right y-axis, that's the microcystin in water, and those are the blue bars that you can see. And then we have the same health-based thresholds. The solid gray is the California action threshold, and then the dotted line is the World Health Organization tolerable daily intake. And a few things that I want to point out here is that in general, it seems that the toxin concentrations in the clams peaked at the same time as the water samples, and then seemed to quickly deprecate back down to background levels. The other thing that's notable is that we did not detect any microcystins in the water samples in 2022, but you still see a spike in a lot of the clams at that date. So perhaps we just missed the toxins by collecting those discrete water grab samples. And then the other thing that is notable is that you kind of see this persistent low level concentrations of microcystin present at all of the sites in the clams throughout the entire year. So this
is in agreement with those past findings in the estuary, but the Peacock and Gibble studies that also found year-round toxin contamination. Although we cannot 100% rule out matrix effects, the LCMS method did confirm the low-level samples as well. And so it's possible that these clams are just, clams are feeding year-round, and so they're filtering water year-round, and so microcystis cells do settle down into the sediment during the winter, and maybe they are filtering that toxic water year-round and having this baseline toxin concentration. And then I also noticed that the Cache Slough area here and here, and then down at the Sacramento River, seemed to have a few more non-detects than the other locations. Interesting finding from the study is, so this is all the sites combined together, and wanted to see if there was a seasonal pattern, and it appears that toxin concentrations are highest in clams from July through November, which lines up with when we see the highest toxin concentrations in the water column. So the next few slides are about communicating these results. So the first goal was, is microcystin a stressor on native fish species? If you remember from the beginning, we said if it's present, then we're going to assume it's a potential stressor because there's no safe or unsafe amount of microcystin for these fish to eat. So there is a potential that these clams are a stressor to fish that are consuming them. Sturgeons have been known to eat Asian clams. It's been found in their gut content, but it's not their preferred diet. So we don't know how many of these clams they're actually eating. We don't know if there are other prey species that are contaminated with microcystin throughout the Delta. And we really don't know how much toxin is a true risk to these fish species. We plan for continued coordination with the DWR and CDFW sturgeon team. And then the secondary project goal is microcystin and shellfish a risk to human health. So we found that microcystins can be present in Asian clams, but average microcystin concentrations are low, except for a few outliers, and do not present a human health risk. We think it's safe to consume Asian clams from our study area during CCHAB events. We suggest that further research on crayfish is necessary before a recommendation can be made for those organisms. It appeared that the crayfish accumulated slightly higher concentrations of microcystin than the clams. I forgot to mention that on the box plot slide, but a t-test revealed that there was no significant difference in the toxin between the two organisms. So we don't think that our data set is large enough to make a specific species recommendation for the Asian clam or for the crayfish. It's also noteworthy that 18 of the 30 crayfish were collected from the Cache Slough site, which had the lowest water column microsystems. So one of the things that outputs that we are developing for this study is a fact sheet for public health professionals. And this is a first draft of that fact sheet. But what we're including in it is the project overview and our study sites, and then the major findings from our project. The two messages that I just read to you from the last slide are included in here. And then this communication strategy for low-risk microcystin exposure. And I have a bigger picture of this framework on the next slide, and I'm just gonna walk through it quickly. So first, this is should back up and say that we developed this based in part on information from the ITRC and other risk communication strategies. And we wanted to consider how it's important to not, draw more concern to this issue than the reality that there was this really low toxin concentrations with just a few outliers of microcystin, high concentrations of microcystin. So how do you present kind of this low risk microcystin exposure? So first is identifying and determining risk. And so we compared our values to these established thresholds. And then considering how to communicate the potential for microcystin risk, being careful to not, emphasize these a few high findings out of 339 samples. And then identifying the intended audience. And so we enlisted some potential groups that this information could potentially be communicated to. And then developing our message, which is that the concentrations of toxins in the clams were low, and we believe it indicates a negligible public health risk. Then hoping to work with public health agencies, and we've been in contact with both OEHA and the California Department of Public Health to start initial conversations about this, and would definitely value their input on our fact sheet before we finalize it. But just figuring out the best way to deliver the message, and then getting feedback after delivering the message so that we can figure out how to improve messaging in the future. So some future research ideas are to investigate these low-level toxin detections that we had in the shellfish. Just like to confirm that there's no matrix effects. Like I mentioned earlier, we don't think that's going on, but we cannot 100% rule it out. We'd be interested in looking at toxins in the sediment to see if these clams are just being exposed to the toxins year-round. We'd like to expand the crayfish sampling, especially interested in looking at areas that have higher microcystin concentrations in the water column. You could also consider looking at shellfish from areas recognized as bloom hotspots, but I put an asterisk there because the use of these hotspot areas by shellfish is unknown. As an example, we've done a lot of work in the Stockton waterfront and collected a lot of sediment samples down there, and we don't see any clam or see very few clam samples in that location. So that wouldn't be an area that I would suggest to study, but there may be other CCHAB hotspots that shellfish utilize. And then I just put in here to stay tuned. We're doing a toxin analysis of crabs and shrimp. This is funded by NOAA MERHAB project, and it's a reach from Pittsburgh to Vallejo to look at if microcystins from the delta are being transported to the bay. Those past studies indicate that may be happening, and so we'll be analyzing these tissue samples. And with that, I'm available for any questions. I'd like to thank everyone that helped with this project. It's been a big effort from all of these folks.
Thanks, Ellen. We've got a couple questions in the chat here, starting with Sarah here. What consumption rate level are you using to determine low health impacts?
So for the OEHA, that was, I don't remember the serving size that they used, but for the World Health Organization, it was a 100-gram meal, and a 60-kilogram person was factored into the calculation. I think OEHA's was a similar consumption rate to come up with their threshold.
Ellen, this is Reggie Linville, and I think that we had a 1 8-ounce meal a week on that work. And I was going to say first of all, I think that was a great talk, and thank you. But I think I would be a little careful about saying there's no risk with this, because that is for moderate intake rates of this fish tissue. If someone were doing a higher intake rate or a subsistence intake rate, it could be a very different scenario.
With the very low, generally low concentrations.
with the concentrations that you showed, if someone say they were eating 2 or 3 times the amount that was factored into the action level, then they would be up into risk levels that are, of concern.
Thank you. Yeah. So you're one of the people that we want to coordinate with on the fact sheet, since you and I emailed about that earlier. And so this like definitely the type of feedback that we value getting, because we're just basing it on those, available thresholds that are in the literature.
Well, it's important to know that moderate intake is not of risk. I think that's really important.
I don't know if I highlighted it, but I also was trying to emphasize that this is from our study locations only and not trying to apply it to the Delta as a whole, because we can't say that based on our study results.
It looks like Sarah has a couple more comments for you on that topic. That maybe it looks like the Mercury program has some intake rates for how much people are eating at different tribes. Anyway, you can read those comments too. But she'd like y'all to coordinate with a WEHA, it sounds like, too.
And we are planning on doing that before finalizing that fact sheet. So
awesome.
Appreciate all of that input.
Okay, and then we got another comment or question from Christine Job. Can SPAT be used in the crayfish areas to provide insights into potential exposure, and then combine the species with rates of uptake by these species?
I didn't go into it in this talk, but this past summer we did deploy SPATs it was with the clams sites though, not the crayfish areas, to start looking into that idea. It was a low toxin year in the Delta, and so we got pretty low concentrations of toxin. So it ended up not being a great comparison, but certainly something that could be looked at further. And for the MERHAB study, we do have SPATs deployed with the crabs and shrimp so that we can be comparing the concentration so that we're not just relying on water grab samples.
All right. Anyone else? Final questions? All right. Well, thank you so much, Ellen. That was really interesting to get those early results. And yeah, I think that'll be closest out for today for the most part. Any other last questions, thoughts, updates? While we're all together. All right. Well, thank you so much all for attending. Our next meeting will be April 24th, and I hope to see you all then, and probably many of you before then. Enjoy the rest of your day.